First of all, let me thank so many of you for making it down on this cold, rainy day to the Harvard Club to hear some information about American Gene Technologies, and in particular our development of an HIV cure. My name is Jeff Galvin. I'm the CEO of American Gene Technologies. I founded the company, and I've been with the company now for 11 years, which probably sounds like we're getting long in the tooth. But I'll tell you that we have taken a unique approach to gene and cell therapy that really has to do with understanding the fundamentals and developing a lot of tools and reusable components, which makes solving problems with gene and cell therapy much more efficient. So what I hope you're going to learn about today is a very exciting development that has the potential to move HIV-infected individuals from lifelong antiretroviral therapy to what we call a functional cure. It's a technical term, but think of functional as being as good as. So what do I mean by as good as cured? Well, it is possible that HIV patients could have very long-lasting viral reservoir. It hides in the body when people have been infected for a while. But the antiretroviral therapy keeps the viremia down in the bloodstream, which is what keeps it from turning into AIDS. And what we're doing is we're replacing that antiretroviral therapy with a powerful fortification of the natural immune system. We are taking HIV T cells and we're modifying them so they're immune to HIV. Well, what does that mean? Well, the only reason that HIV T cells don't clear HIV from the bloodstream in the sort of average person is that they can be infected by HIV. They can literally be part of the chronic infection. When an HIV T cell comes along to eat a viron, instead the viron grabs tightly onto the HIV T cell and it merges with it and installs the AIDS genome, the HIV genome, inside that cell. And for the rest of the life of that cell, it becomes part of the problem. And in fact, it's the HIV-specific T cells that run to the infection that are the first to be infected and the first to be depleted. It's ironic, but elegant in a way. And I think you're going to find this irony persists in the scientific community that, as hated as HIV is, it's quite admired for its remarkable evolution and what it's been able to do. Well, it turns out that a relatively small change to those HIV T cells that is backed up by decades of research. So we know what it takes. We have to do the experiment to see that this works, but there's a lot of information out there that explains to us how you can modify that T cell so that it can't be infected. If it can't be infected, it clears HIV like cold T cells clear a cold, like flu T cells clear a flu, like bacterial T cells wipe out bacteria. That is the whole idea behind AGT's HIV Cure project. It's just to restore natural immunity to HIV. At that point, once we have done that, then your HIV T cells will control any HIV in your body. So HIV-infected individuals, even though they might have a little viral reservoir, it's almost as if they were it's actually more effective than taking antiretroviral therapy because their body will naturally clear those virons. And over time, they'll clear the viral reservoir as well. That person will be HIV-free. We don't know right now how long that will take, but theoretically, that will happen. But in the meantime, the important thing is they cannot progress to AIDS. So they are protected from AIDS. They are not infectious. They're not contagious. They cannot give this to anybody else. And here's the beauty. They can never be reinfected. Because once you're immune to a disease, like the flu or a cold, you can't get it again. So this is remarkable. They're better than cured. They're immune. They can go back to even high-risk communities without the worry of being reinfected. So it's a tremendous difference, but the most important difference I think, will be a personal one for all of those HIV-infected individuals. And that is that antiretroviral therapy, for people who are not in that community, for the people that are not scientists in this area or doctors in this area or are actually HIV infected, they don't realize that chemotherapy is highly toxic. That daily pill comes with it a lot of the symptoms, a lot of the side effects of chemotherapy: nausea, headaches, loginess, dizziness, fatigue. And long term, it leads to early aging, bone density issues, osteoporosis, brittle bones, liver, kidney, and heart disease, and extra cancers. So HIV-infected individuals are far from cured right now. They're far from living a normal lifestyle. And that's one of the things that's the most exciting to us at AGT, is we think we have the opportunity to make a profound difference in 1.1 million American lives alone, and 37 million people globally. So out of the chute, I think what we've created is a commercializable HIV product for the Western world. But again, going back to the history of AGT, what we've been doing is building tools and components to make it easy to develop new solutions for patient problems on gene and cell therapy. Reusable components that you can mix and merge together that give you access to broad disease areas. And so I hope what you're going to find out is that HIV is just the first cure, the first product off of this platform, with many more to come, and that the difference between AGT and every other gene and cell therapy company boils down to one word: efficiency. That we have become the most efficient software developers for the organic computer, the human cell. And that will give us a unique opportunity to not just provide solutions like this HIV program that you're going to hear about today, but to turn out better solutions, to actually focus on not just opening up new areas with this, but taking existing areas and essentially obsoleting ourselves. That coming out with better and better solutions when those solutions become possible. So think of this as the software industry for the next 100 years, reprogramming the human computer, DNA, and the human cell for better health. Viruses have been making us sick or modifying our DNA for 1.5 billion years. The recent development is that we can crack open these viruses, scoop out the viral DNA and throw it away, and we can use their carrying capacity to bring in modifications to our genetics that are beneficial and improve our health. Essentially, what this industry is doing and what we're doing, and what we're doing I think more efficiently than anybody else, is converting viruses into updates, right? Updates for your cell that can either replace a gene that's broken, that would be things like hemophilia and beta thalassemia and phenylketonuria, that's one we're working on. Improve the operation of cells. That's all that the HIV product is. We're just improving HIV T cells just enough to cure somebody. Or even modifying cells to make them little bots in your body that can do anything. And an example of that, which you've all heard of, is CAR T. By making a modification to a T cell, they've sent it off in a totally different direction. This shows you the scope of what can be done by repurposing these viruses, by reprogramming these cells. We're on the eve of a very exciting revolution in pharmaceuticals. AGT hopes to be a leader in this industry for the years to come. We've got a patent portfolio and IP that's been valued at $2.4 billion that forms this platform of tools and components that can be mixed and matched for a myriad of solutions. What's that sound like? sort of sounds like the MS-DOS of this new computer industry. I'd like to say that we're the iPhone and curing a disease is just like writing an app, but that's the dream, that's the vision. And you'll find that it's a unique vision within this industry. Anyway, I'm excited to hear from our panel of experts. Today, I think you're going to get a really good education in the existing developments in HIV and the history of it. And you're going to get a really, from the horse's mouth, you're going to learn how AGT is addressing this problem. I have been very lucky in this business. I've worked hard, and I had a vision and I had a dream for this thing. But in running around and talking about my dream of building this kind of company, I have attracted some of the best scientists in the world. And this is something that I am incredibly proud of, and I got to tell you, I'm really excited to hear from 3 of them today. So I won't hold up the main program any longer. I want to introduce our CSO, David Pauza. David came to us from, most recently, the Institute of Human Virology. I'm going to let him introduce himself as much as he would like to. But one thing I want to tell you is that was a real turning point in the company. When Bob Redfield, current head of the CDC, who was one of the directors of the Institute of Human Virology and one of the founders, called me on the phone and said, you should hire this guy. This guy was his right-hand man, his scientific pillar at one of the top research institutes in the world on HIV. And Bob Redfield believed that by putting the 2 of us together, we would have all of the elements that we needed to really make a difference. He actually said to David, I think you have more chance of curing HIV down at AGT than at the Institute of Human Virology, which is high praise. I mean, that is a source of tremendous research and treatment technologies. But that was a big turning point, and I got to say that I've been lucky to attract people like David and the 2 other scientists that you're going to hear from today, Bill Blattner and David Hardy. And this is just a sampling of a few dozen people in all different areas that have been attracted to that idea of using gene and cell therapy as a powerful new technology for solving problems. And I think that that was sort of a turning point for the company when we really formalized everything that we had, and we got 3 things really nailed down and in process. First one is HIV. That's the first one going into the clinic. You probably heard that we already submitted the IND on that. We have a cure for phenylketonuria. That's to prove out our monogenic platform. And then we won't get a chance to talk about it today, but I hope we'll bring you back here again sometime for a talk on a unique immuno-oncology asset that is already getting a incredible response in animals in a 1,000 mouse study of advanced solid tumors of epithelial type. We're getting 85% complete remission with 1 shot of a viral vector to the primary tumor. And this should translate beautifully into humans. But anyway, that's that is all about our approach. It was to build out all these components and then to figure out what's the low-hanging fruit where we can really make a difference. And I got to tell you, we've got a ton of that stuff cooking back at the company. And I think after you hear from these guys today, you'll understand how serious we are about this stuff. So anyway, as I was saying, I was very lucky to attract David to the company. He's been working with us for over 5 years now. And proud to introduce him right now.
Can you just click on the agenda slide there?
Sure. There
So
we go.
I'll just say good morning, and I'll say a little bit about my background later. This is the plan for today. This was the optimistic time schedule, but we won't get too far. We won't stray too far from it. And we've invited today 2 of my colleagues, my academic colleagues and friends in this field of HIV, Both of them have really kind of a lifelong commitment to this disease, as do I, in fact, personally. And I'll first just say that our first speaker will be William Blattner. Bill was a longtime member of the NIH and was a pioneering investigator in a number of human viral diseases. He's an oncologist, but he also became very interested in epidemiology and public health. Bill has been involved both domestically and internationally at a very high level in combating HIV worldwide. Bill is also a co-founder of the Institute of Human Virology in Baltimore, Maryland, and now is running a private company in Florida. David Hardy is going to be our second speaker. So Bill will talk about the antiviral therapy and what's been done in that space to try to approach a cure for HIV. And I think that it's valuable to know where we are currently with the development of antiretrovirals, and now what's coming along that pipe besides very small molecule compounds. What are the biologics? What are the other considerations going into what kind of progress is being made? David Hardy has a long-term interest in research, gene therapy, and clinical medicine for HIV. He was the chief medical officer at Calimmune, which conducted an HIV gene therapy trial in Southern California with a number of clinical trial sites. And he's also been a very dedicated physician for HIV with a lot of contact in the community and a lot of knowledge about how things, what things are really perceived of value and what are the best ways to go forward in this program. So David will introduce you to the area of gene therapy for HIV, which is actually much larger than most people understand. It's probably the single largest area of gene therapy studies that exists. And it's really kind of unknown to some degree, partly because most of those studies were done by academic institutions and very few of them done by companies. But David will give you an overview of that and sort of read you into that whole area, and also then talk a little bit about community perspectives and what people are looking for from us for HIV cures, and what is the appetite for cure out there in the HIV positive community. I will then follow up with a very brief discussion, just a little bit about the characteristics of our product to show you how we learn from the gaps in the literature and the gaps of what other people have done and tried to fill them. So without saying anything more, Bill, please, if you could take the stage and talk to us.
This is a great opportunity to get to know you all. I'm very excited to be involved with American Gene Technology. It's really a tremendously innovative approach that's being developed. And so I'd like to spend a few minutes going over where we are with the current therapies that have been developed over the years and look at the prospects that the current treatments could result in a cure or not develop that way. So the HIV virus is a lentiretrovirus. It's got a membrane. I guess this pointer doesn't show up on the screen, so we don't have to worry about that. I like to walk around, but I guess I'll be tethered here. Okay, so I can point at my computer here. So anyway, it's a on the left, on this side, on the right side is the picture of the virus as it looks under electron micrograph, and on the right is a schematic that shows the major components. And so on the outside of the virus is something called GP120, which is the major envelope protein of the virus. All of these are targets for different therapeutic approaches, particularly the GP120, which is a focus of biologic targeting with antibodies. The reverse transcriptase at the bottom is a protein that is made by the virus that is involved in taking the genetic material of the virus, which is an RNA genetic material, and it's able to reverse transcriptase that material into DNA. Okay? So this is reversing the normal process of biology. The normal biology is the cell has DNA, and then it makes RNA, and RNA makes protein. Well, this virus turns that upside down, and it takes the RNA and makes it into DNA. And that's a target. That enzyme is a target. The second step in this process involves the integration of that now new viral DNA into the genome of the host. So it's like cancer. You got a gene that gets put into the cell, into your genetic material, and that's why cure is so difficult. And so there are recent drugs that attack that gene, the integrase. The protease was the second target for antiretroviral therapy. And the protease is involved in taking the proteins of the virus, cutting them up so that you can make another virus. So that's sort of the basic introduction to the virus. Now I'm going to show you a little bit about the biology of how the virus gets into the cell and how and what that involves. So this is a sort of lunar landing look at the virus as it's coming in. The cell is here on the bottom. These little blue things are what we call the chemokine receptor. You'll be hearing a lot about that. CCR5 and CXCR4, the name of those things. You'll hear about those later because they're important to the technology you'll be hearing about. And then those orange things are that chemokine receptor. The blue is the CD4 receptor. So this moon thing comes down. It gets tethered like a ship coming into the harbor with a small line that attaches the CD4 receptor. And then it finds that other receptor, the chemokine receptor. And during that process, there's a conformational or shape change to the GP120. And that's important because it exposes new parts of that GP120 that allow new antibody targets to be identified. And so as you hear more about some of those therapeutic things, you'll be hearing about some of those antibodies. So then the next thing that happens is that the virus has a syringe that shows up. Once that attachment takes place, then the syringe comes in and it pops a hole in the cell. And then the genetic material flows into the cell. Okay? And now I don't know why this thing is going automatically, but anyway, we'll have to go back. So now we're inside the cell, and we have the genetic material in red coming in here. The reverse transcriptase is making a DNA copy. That DNA copy becomes part of the genetic material of the cell. And now you are infected for life with this virus, and you're never going to get rid of it because it's part of the genetic material. And that's why the cure has not happened yet. Okay, and so at each stage of this life cycle, there are drugs that I'll be telling you about that target here at fusion, the reverse transcriptase, the integration, the assembly with the protease, and with the budding. And so each of these areas of that life cycle of the virus also presents targets for some of the drugs that are currently available. Okay, so what are some of the important features of HIV/AIDS? Well, its transmission occurs via contact with virally infected cells in the genital fluids or with free virus in these fluids, blood or other tissues. So the virus can either be in plasma or in semen, or it can be in the cells that are occurring in those tissues. But it doesn't occur by casual contact, by shaking hands, kissing, and that sort of thing. It's not a highly contagious virus. The principal target, as I mentioned, is the CD4 cell. It attaches to the cell, as we showed, via that CD4 molecule, and subsequently uses this chemokine receptor, CCR5, which is one of the targets that you'll be hearing about in this therapy that's the showcase for today. There's two types of the chemokine receptors, the CCR5, which is most common, but also this other one called CXCR4. There'll be a quiz at the end of the presentation. The result of this is that this virus kills off CD4 cells. And what do CD4 cells do? Well, they're the major part of your immune system. And when you kill the immune system, you kill the host. Now, the immune system is not just these little lymph nodes that get swollen up sometimes when you get a sore throat. The immune system is throughout the body. If you're going to prevent infection, you have to have immune cells throughout the body. They're lining the gut, they're in your mouth, they're in every part of your body. So the immune system is a huge organ, but you can't point to it like the heart or the liver or any other part of the body. So it's throughout the body, and it's able to mobilize itself and go to where infections are occurring and so on. So the current treatments slow the disease progression and allow a near normal lifespan, but it does not eliminate the virus from the body. And as you heard earlier, there's side effects and there's a lot of other issues that we'll go into. Because HIV becomes part of the DNA of the cell and often goes into hiding in reservoirs, which you'll hear about in a few minutes, current treatment does not cure HIV. Therefore, treatment requires lifelong daily pill taking associated with adverse drug reactions, organ toxicities, resistant viruses, and high treatment and healthcare costs. And one estimate that's published, and it may be a low estimate, is that the lifetime cost per person is $367,000 and change per person. That's a huge healthcare burden for the lifetime of the person. So this gives you a little bit of a picture in a capsular way of the devastating effects of the HIV virus. This individual on the right had severe immune deficiency and was on death's door, and through the PEPFAR program, which was an international global program that made antiretrovirals available widely in Africa, he's now restored to health. He's a father. He's a productive member of society. And so the drugs have had dramatic positive benefits. It was horrible in the early stages of the epidemic to go to the hospital and be with patients who were dying a very painful and horrible death. But then in around in the late 80s, we had our first antiretrovirals, but they were single agents, and the virus quickly escaped from those drugs. And then by 1995, there were multiple classes, the protease inhibitors and the reverse transcriptase inhibitors, and you ended up with a handful of pills. I mean, you were taking 20 pills a day. Some of them you had to take with a meal, some without a meal, some at bedtime, some in the morning, and you had to take them at very specific times because the pharmacokinetics of these drugs were poor. And therefore, if you didn't take it at the exact time you were supposed to, the virus would pop back up and it would become resistant. Today, it's a single pill. This is a remarkable advance in science over these years to be able to move from nothing to a handful of pills to a single pill. And now we're going to the next generation for a cure. So these are the classes of drugs that are available currently. If you go back to that slide in your mind of the virus coming in and then going through the cell and so on, each of these stages show where those drugs are occurring. So there are drugs that impact the binding of the virus over here, and some of them are antibody and some of them are small molecules. Then the reverse transcriptase inhibitors, there's two types of those that have been developed, and they're sort of the anchor treatments. Then there's the integration inhibitors, the integrase inhibitors, and then the viral protein processing drugs, the protease inhibitors, and then recently in the pipeline are drugs that are targeting the assembly of the virus, the viral capsid assembly drugs. And there's one from GlaxoSmithKline that is actually in clinical trials and has shown good antiviral effects, and also has some advantages from the point of view of being a longer acting type of drug. And we'll be coming into that a little bit later in the presentation as one of the strategies. So the goals of antiretroviral therapy are to reduce the morbidity and prolong the quality and duration of life, to preserve and restore the immune function, which is vital to life, and suppress the virus. And one of the benefits of this is that you also prevent infection. And in fact, for those of you who may or may not have heard of it, there's something called undetectable, uninfectable, the U equal U. You may have seen in some billboards and stuff like that. It's the concept that if a person who's infected is having sex with someone else and they're virally suppressed, they won't transmit the virus. And there's also a treatment called PrEP, which people can take an antiretroviral if they're going to have sex with someone that might be infected. But we won't go into that. That's not the focus of today. So there's a decision-making process that physicians are involved in. When to start? Well, the current idea is let's test and treat. Let's get everybody on treatment as quickly as we can. Not worry about the viral, the CD4 count and that sort of thing. But there are other nuances to this. Is the person going to be able to take the drugs? Are they high on crack, etc.? These are all things that can influence your ability to take medications. If you're stoned out, you're not going to take your meds and you're going to develop a drug-resistant virus. Also, there's issues of pregnancy and other comorbidities that I'll touch on a little bit later. And also, some people receive a drug-resistant virus when they have sex. They have a partner who has a resistant virus, and they start taking drugs that they're already resistant to. They ain't going to work. And those guidelines are published by the Public Health Service. So the sort of first-line regimen for most people, and I won't go into details on this other than to say that the current regimens involve an integrase inhibitor and a nucleoside backbone of reverse transcriptase inhibitors. So those two drugs together are highly effective and well tolerated and have good pharmacokinetics to work. And there's two options that are shown here. And then there are other combinations that are also used, but in special situations, and I'll touch on what those special situations are in a moment. Some involve protease inhibitors with nucleosides, others non-nucleoside reverse transcriptase inhibitors plus nucleoside backbone. Many of those are being used in the developing world. And then the integrase inhibitor plus a nucleoside backbone. So these combinations of drugs have gone through clinical trials and have been shown to be highly effective. So what are some of those special situations? There are factors that are associated with how sick the person is, whether they have a very low CD4 count, a high viral load, whether they have certain genetic factors in their background, whether they might be having a drug-resistant virus, other issues related to the characteristics of the drugs themselves in terms of single pill formulation versus multi-drugs, food effects, and then particularly important are the bottom three checks, so to speak, or four checks, the other conditions, underlying kidney and liver, bone, psychiatric, HIV neurologic disease, cardiac disease, etc. These all can play a role because of the effects of the drugs on some of these organs. Pregnancy also impacts what you're going to take. And hepatitis B and C, there's interactions there, and TB. So the point here is that this is not your basic doc in a box that is going to be making all these therapeutic decisions. This is pretty complicated stuff. And so this is not just some simple, we're going to give you an aspirin and call us in the morning. This is something where you have to really know what's going on with the patient to tailor the therapy And so it adds to the healthcare cost and to the burden, and promotes the concept of cure would be a nice concept. So, moving on, the treatments that have been developed are miraculous. People are living, and they're living normal lives pretty much, but not completely normal. But it's not the long-term solution. Patients are at ongoing risk for major comorbidities. These drugs and the underlying HIV contributes to cardiovascular disease, diabetes, kidney disease, and HIV is an inflammatory virus, and so it's damaging your body constantly, even though it's suppressing the virus. There's issues of adherence, drug resistance is a concern, and the estimated cost, as I pointed out, $367,000 per patient for 1 million requiring antiretrovirals. In my opinion, cure, although costly, I mean, these cell-based therapies are expensive, as you've probably read in the paper, but they may be economically feasible as an alternative. That if we're spending this much money, maybe a single-time cure that costs hundreds of thousands of dollars, in the long run will have greater benefits, not only in terms of curing the disease, but also improving the health of the individual. So that's part of the calculus to put in. So you heard earlier about the reservoir, and this is just a cartoon to kind of show what that concept involves. So at the time of infection, very early in infection, HIV establishes a latent reservoir, which means the virus is genetically integrated into the DNA of the host, into your DNA, and then it lies there and hides. Okay? And the red and blue parts of this figure show the virus that is replication competent, which means that it can pop out of latency and reinfect the person if you stop drugs. The yellow is the large pool of defective virus. The reverse transcriptase is not a very good enzyme, and so it makes a lot of faulty copies of HIV, which cloud the picture and contribute to that concept that you heard about, that you may not have an eradication cure, you may have a functional cure. You may be able to detect virus, but the virus that's there may not be functional virus. So anyway, during the course of infection, you can see a growth in the amount of virus in the body, and then once you start treatment, then you kind of freeze that pool, and that is one of the major issues that you confront. Now, there are clinical trials that have been going on where we're trying to see, does this intervention result in a cure? The first type of that was, well, we've got the virus suppressed, maybe we've gotten rid of it, let's stop therapy and see what happens. And so when treatment is interrupted, the latent reservoir of cells containing the intact virus, which are red and blue in this, reignites high level of viral expression. And this reignition occurs even after decades of being on the therapy. So if you're on therapy for life, you still have that latent reservoir hiding in your body. So HIV drugs can suppress the replication and decrease the reservoir, but they can't eradicate the virus from the body. So there's been a lot of studies, over 100, that we put together, and I won't go through all of the list of things. You'll be hearing a little bit more about these. But basically, there's a whole host of cure strategies, and you'll be hearing in more detail about some of the newer ones involving gene therapy a little bit later. But they involve a variety of treatments with antibodies, with inhibitors of inflammation, gene therapies, things that go in and alter the cell signaling, high levels of treatment, etc., gene modification. These are all approaches that have been tried, and we haven't gotten any cures from those. But I'll tell you a little bit about a couple of patients who've been cured in a few minutes. So just to reiterate, the cure definition, there's sort of two definitions. The eradication, that's not going to happen. That virus is hiding in there, that defective virus. You're going to always be able to detect some virus in the cell with that latent pool. But HIV control, the functional cure, is feasible. This is a cure where you might be able to detect virus, but you don't need antiretrovirals because that virus doesn't come back. Your immune system is fully functional, and the virus is not having adverse effects on your body. So that's the functional cure that is really the target that I think is feasible. And so in terms of the HIV eradication efforts, there are latency reversing agents. The idea was we're going to put in a drug that stimulates that latent pool, and we're going to put the drugs that treat the virus, and we're going to cleanse the body of the virus by flushing it out. Well, that hasn't worked, okay? Antibodies, I'll tell you a little bit more about that, but there is some evidence that targeted antibodies that are, have good broad neutralization do have some effects. The stem cell transplantation you've read about in the newspaper, you know, headline New York Times, Berlin patient cured, London patient cured. Well, the background is there's been 43 people who've gotten this treatment. Okay, what does that treatment involve? It involves a bone marrow transplant from a donor that's not you. And the donor that you receive that donation from has a genetic defect called Delta something or another, 32. It blocked my mind here for a minute. But basically it means that the CCR5 receptor is defective, and therefore the virus cannot infect those cells. And so the idea was we'll give a transplant with that missing gene in it, and we'll cure them. Well, they tried it in 43 people, and 2 succeeded. And the reason the others didn't succeed is that they use a CXCR4, the other chemokine receptor. And so, and these patients, if you've ever been around a transplant patient, it's not a walk in the park for sure. And then gene editing proposes to remove the CCR5, but it does not address the existing reservoir, and then that issue of the CXCR4 virus emerging. It's a very malleable virus. Okay, so how can we achieve ART-free remission of HIV infection? We can achieve viral control without ART, but requiring intermittent or continual non-ART intervention. Or we can achieve an ART-free remission by inducing a durable immune response to control the virus. So in one case, you're doing intermittent therapy, but not every day. And the other, you're controlling the virus without further intervention. So there's been a number of approaches that have been attempted: therapeutic vaccination, passive antibody transfer, and passive T-cell transfer. So this is the results of a clinical trial, and it's pretty obvious that the placebo and the vaccinees showed no benefit. So basically what you do in this situation is you have a person on ART, on treatment, you vaccinate them with a vaccine to build antibodies against the virus, and then you stop the treatment and see if the virus is controlled, and the virus is not controlled. That's sort of the standard protocol for a lot of these studies. You do an intervention, and then you stop the antiretroviral treatment and see what happens. Okay, so that showed no benefit. What about passive antibody transfer? Well, this is a picture of the GP120 in 3D. It's a pretty complicated looking molecule. And the main point of this is that there are a lot of different antibodies to a lot of different parts of this GP120, that outer membrane protein. And many of these antibodies are what we call broadly neutralizing. They're able to neutralize many different strains of the virus. One of the important things to understand is that hopefully everybody in this room has had their flu vaccine this year. Okay? Why do we get a flu vaccine every year? Because the flu virus changes. Well, if you look at HIV, it's about 1 million times more variable than the flu vaccine, the flu virus. Okay? It is just incredibly malleable, and therefore single antibodies just are very unusual that can neutralize more than one type. Okay, and so this is a trial with single different monoclonals that were developed that had broadly neutralizing ability, and the same protocols followed. You infuse this antibody, and then you stop the antiretrovirals, and you see when the virus comes back. And you can see that
Actually, HIV.
to 80 days, some people went out a couple of weeks, and so there's quite a range across the different antibodies that were given. But they did show some effect, that you could suppress the virus for a period of time, but then that effect wore off. And so this is a potentially promising approach, but it's not going to cure. It's going to help maybe prolong the ability to suppress virus. So there's efforts to look at more potent antibodies, extend the half-life of the antibodies. There's studies to try and create vector-based antibody production and combinations of antibodies, which is currently in trial. And these two studies published last year showed that the combination of multiple antibodies had some significant benefit in prolonging the suppression of the virus up to 21 weeks, but on maybe 3 months, whatever. But the main point is that the combinations of antibodies may add to the ability to suppress the virus and may help us move towards having less frequent need to take antivirals every day, for example, which would be one of the goals. And so passive antibody therapy results in transient viral control. There is the problem of viral escape, because there's those latent viruses sitting around and they adapt to the environment that they're in, and combinations of antibodies do have some benefit. So the final focus of this is the current focus of the pharmacy industry is to develop longer acting antiretroviral therapy, so you don't have to take a pill every day, for example. Because treatment will not cure, efforts are being made to make the drugs less needed every day. And so they're being formulated in various slow release approaches using nanoparticles and other strategies and other devices that might be implantable. Cabotegravir is being developed by GSK, and it's an integrase inhibitor that has favorable properties for long acting. It's being looked at as a potentially monthly type of dosing, and it's being co-formulated with another non-nucleoside reverse transcriptase inhibitor. It has good chemistry for this, but the problem is that in the yellow bullet there, you have to give separate injections of these two drugs to get the benefit that you're looking for. So, in summary, we have potent, the first point is that potent, reliable cure therapies have not emerged from these multiple hundreds of previous trials, and the trials that are ongoing are increasingly complicated, combining a variety of modalities. Therapeutic vaccination has not benefited. Intermittent antibody therapy provides some benefit and may help to decrease the need for daily treatment. That's an expensive approach, though, as you can imagine, from other monoclonal antibody therapies that you're probably familiar with that are being used for a variety of diseases. And there's some long-acting antiretrovirals in the pipeline. The bottom line is we're not at a point where antiretroviral therapy can result in a cure, and we need to move to that next stage of how we're going to eliminate the virus through the cure strategy. questions or anything that need clarification?
Well, we'll have a question time at the end. So David, if we can call on you now to talk about gene therapy.
Thank you, Dr. Blattner, for that wonderful introduction and setup for what I'm going to say now. Just to give you a bit of a background, I started my career in HIV in 1982. I saw my first patient with HIV in Southern California as a trainee, and went to look for the guy who discovered reported the first cases at UCLA and became his fellow, and have been doing this pretty much my entire life. Career-wise, I always wanted to do infectious diseases, and research was always something I liked, and so HIV research really came along very nicely. I got involved in antiretroviral therapy very early, gave AZT to some of the first patients in the country, in the world, and then involved through the different sort of clinical trial groups, the development of antiretroviral therapy for several years. Until 1990, this is all done in Southern California at UCLA. In 1996, a big thing happened. And that was that we were able to actually treat the disease effectively with oral medications, because we put 3 drugs together. And that was a wonderful thing because we could actually start seeing patients no longer die, but live. And it was actually almost a day you could say this stopped and this started. And it was wonderful. By 1998, I was actually getting kind of bored because things weren't as exciting and challenging, because everyone was doing great, which was fantastic, but some of the challenge of taking care of people who used to be very sick was changing. So I went into a laboratory because I wanted to learn more about what HIV was all about. And the people, for the first time, started using the word eradication or cure. And I got in the lab and worked with a basic scientist, a molecular virologist, for 4 years, learned how to make viral vectors, learned more about HIV, really understood how this little virus is very diabolical and very, very well organized to do things against our human immune system that has probably evolved to where it is now. And it also created a great respect of the virus for me, and helped me better understand that curing HIV can and will happen, but it's not going to be easy. It's not going to be simple. It's going to require some very innovative thinking and some very new and different approaches to how we look at cures. And so I got involved in my first cure research as a postdoctoral fellow, and then ran my own lab creating viral vectors for about 12 years in Southern California, and then worked, as Dave mentioned, for a small little company. My lab mentor, Irvin Chin and David Baltimore, teamed up and put together a small company called Calamune, funded primarily by the California Institute for Regenerative Medicine. And they wanted to translate their laboratory work into human beings. So we ran 2 phase 1 trials of doing gene therapy with T cells and CD4 cells. And stem cells, CD4 cells and stem cells. And ran a trial doing that. And it really gave me a good idea, a picture of what cure is going to look like, I think one of the most important things that I can leave with you today, talking about gene and cell therapy, is that you kind of have to reset your mind in a way that this is going to be something not like small molecule therapy. Small molecules will be involved along the way, definitely, but this approach is something new and different. And quite frankly, we are borrowing from a successful research now translated into therapy system from oncology, where they have cured difficult to treat cancers like leukemia, lymphoma, breast cancer by doing stem cell transplants. When you really think about it, HIV has really undermined the human immune system in a very specific and very critical way. It has specifically picked out by its tropism for the CD4 cell molecule on the T cell, a specific type of T cell, which many people, including Tony Fauci from the NIH, one of the best immunologists in the world, has called the T cell the conductor of the immunologic orchestra. And when that conductor gets hit by a viral, in this case a foe, the rest of the orchestra can't play well. It kind of does this, but it doesn't really play well. So this is where I think the diabolical part of HIV comes in. That cell was hit very purposely because it controls so much of the immune system. Trying to expect medications like what Dr. Blattman just talked about, to reverse what that is doing is really not reality. We're not going to reverse this with pills at all. We're going to have to do something very, very, I think, in my mind, innovative, disruptive, different in order to cure HIV infection. And this is the beginning of it. This is the beginning of what's really going on here to make this kind of approach work. So I'm going to talk a little bit about cell and gene therapy for HIV, kind of just give you an overview of where this is. Dr. Blatter gave you some very nice basics about how HIV infects a cell. And this cartoon basically shows that HIV uses two receptors. It uses the CD4 receptor that identifies this T cell as a CD4 helper cell, the conductor of the orchestra, very specifically. And it also uses a CCR5 chemokine. So it's sort of a two-step process to be able to, for the virus to attach to and unlock an opening to get into the cell. And so it's an attachment here, and once that occurs, that process happens where the virus basically pulls itself into the cell and injects its genetic material. And at that point, infection has occurred. So how do we best be able to stop that process? Well, we've learned through some small molecule treatment that we can hit many of the enzymes that HIV uses to reproduce itself, such as reverse transcriptase, such as integrase, such as protease. These are all virus-specific enzymes that we can hit and stop that viral life cycle at several different points. And if we hit more than one of those points at the same time, then we can actually shut down active replication for quite some time, hence effective antiretroviral therapy. But the problem is that in order for those medications to work, the virus must be actively replicating. It must be actively replicating. So what happens when the virus is sleeping and is latent inside of a cell? Those enzymes aren't working, so our medications don't work either. And so in order for us to be able to actually get into that latent reservoir of that pocket of virus inside of cells that is no longer going to be responsive to antiretroviral medications, we've got to do something new and different. Now, what happened with the Berlin, the London, and I'll just mention it now, what's called the Dusseldorf patient, because there's now been a third patient that was reported last March who was not off antiretroviral therapy at the time he was reported, but has gone off in the past 3 months and has stayed HIV uninfected off antiretroviral therapy. But it's only been a short period of time. The Berlin patient is about 12 years out off of therapy, so people feel very comfortable with calling it a cure. The London patient is over 2 years out now and is doing very well as well, although hasn't had all the biopsies and careful checking in all parts of his body like the Berlin patient did. And the Dusseldorf patient just went off medication in May of this year. So there's 3 times that it worked, it looks like, very well. As Bill told you, there were 43 times it was done. 3 times it worked. Those other 40 people either died because of the fact that the procedure itself is very, very dangerous and creates a period of time when a person has no immune system and is highly susceptible to infections, or they had what's called a X4 virus. They had a virus that uses the other chemokine receptor besides CCR5. And so blocking CCR, or not having CCR5 here, doesn't really take care of the whole viral infection problem, because there's another pathway the virus can use to get into the cell. And so this kind of procedure, while it cured the cancer, the lymphoma or the leukemia very well in these guys, and cured the HIV, it looks like in at least 2 of them, it's a tough procedure to get through, and certainly not ever going to be scalable. But what we can learn from this is the fact that by blocking CCR5, in this case done by using cells from a special individual who has no CCR5 in their body genetically, normally, we can imitate this by making CCR5 less functional in everyone's cells. And that's really where gene therapy comes in. Trying to extrapolate from the Berlin and London patients, and now Dusseldorf patient, into using that pathway of CCR5 blockade, what we call downregulation, making it less available for the virus to use. If this green structure is no longer available on the surface of a T cell, the virus cannot get inside that T cell. The second step and binding and entering of turning that key to get inside is no longer there. So this is what really underlies a lot of the gene therapy of what we've been trying to use for HIV gene therapy for quite some time. The strategies that have been used in the past have really been many. In addition to either deleting or altering CCR5, we call it either knockout or knockdown by being able to identify it on a chromosome. And like what the zinc finger nuclease does, it grabs a section of the DNA where the CCR5 in the human DNA is, clips that out or makes it dysfunctional. The problem sometimes with that is that we all have 2 chromosomes of everything in our body. So you don't do it in just one chromosome to make it effective. You have to do it in both chromosomes to make it effective, because we have dual copies of all of our genes in our bodies. And if the zinc finger only hits one of the 2 copies, the other one's function still fine. So knock out can be kind of tough, and that's why knockdown may be better in terms of down-regulating CCR5. This is done at the messenger mRNA step, where the DNA is already turned into RNA, and there's just one copy of mRNA of that CCR5 that will eventually make the protein CCR5, but you can target that much more easily because there's just one copy of it in the cell's cytoplasm. And short hairpin RNAs, micro mRNAs, ribozymes have been able to do that kind of thing. Antisense, trying to create an exact opposite copy to the RNA and block it in that way, is also one has been a strategy that has been tried. There has been different kinds of short hairpin RNAs and microRNAs against other HIV regulatory genes that are important to be able to have available for the virus to be able to function normally. CAR T, or T cell receptor T adoptive transfer cells, have been tried as well. NK cell therapy and alpha beta T cell therapy are something that have been put on the drawing board but not put into human beings yet to really see how well that's going to work. And then combinations of different kinds of therapies have also been used, because what we've discovered is that, for example, an anti-CCR5 by itself may or may not be enough, because we know there's 2 different kinds of virus, although one is much, much more common. CCR5 virus predominates at about 97-98% of all viruses in the world today. CXCR4 virus usually 1% or 2% at most. So the viral vectors, how we get the genes into the cell, are also very important, because the whole other thing that really fascinated me about gene therapy when I first started studying it is the ability to really, what we used to call, tinker with DNA. Change the DNA in someone's cells in order to make them better. Now we do this with diseases now, like immune deficiency diseases, where perhaps one gene is missing or has been somehow perturbed. And being able to replace that gene in enough cells has been able to make some of the the congenital immune deficiency diseases treatable, even curable. If you can actually replace the defective gene in enough of those cells, the immune system will come back with enough function to be able to protect the person. But how do you actually get those genes in there? That's one of the beauties that I think retroviruses initially and later lentiviruses really give us an opportunity to do that. Because one thing that people may or may not know about what retroviruses and what lentiviruses do, they're really very similar in their function, is that they basically take genes and insert them into a human cell, which is sort of the most simple and straightforward way of trying to do gene therapy. Except for you can alter a lenti or retroviral vector to cut out the part that you don't want the virus to put in, its own self, and actually engineer a vector to do the same thing, but put the genes you want the virus to put into the human DNA. And so through that kind of elegant molecular biology that many companies like AGT has been able to do, and others, the beauty of being able to add genes or block genes into a human genome is really getting much, much better. There's been an evolution, I think. First, retroviruses were used because they do the same kind of thing, but they oftentimes carry along with them some potential cancer-causing problems. Over time, lentiviral vectors have been used more and more, and there's been at least 3 generations of these vectors developed in order to make them safer and safer and safer and have as little true viral material in that vector as possible. The bare minimal parts of a third generation self-inactivating lentiviral vector just has the parts of the virus that you need to get inside the cell, insert the genes, and then basically die, have no more effect. And that's kind of a really great way to be able to, especially in the case of HIV, use an HIV-like virus to treat and cure HIV. Use something that it does itself in a very diabolical way to get rid of it, to fortify the immune system in a way that we can actually build new immunity, give these cells protection against the virus that in fact is trying to kill those cells. Naked DNA, naked RNA, which is a way to actually also put RNA into cells, has also been tried as well. So these means of actually transferring genetic material into a human cell can be done in many different ways. Adenovirus has been used also many times, but the thing that's different about adenoviruses, of course, is that it's non-integrating, and it's a transient, it's called transient infection. Adenoviruses only carry their payload of genes into the cytoplasm, not into the nucleus of the cell where the DNA is, but into the cytoplasm, and it remains there for a period of time. But if that RNA is used up over time or not replenished, the effect of that goes away. So oftentimes with adenoviral types of infections, because it's not a permanent sort of integration into the human cell's genome or DNA, it has to be repeated to give the cell another dose of that new DNA that you want inside the cell. So some of the critical steps in the manufacture of gene-modified products, because now we're not talking about small molecules, we're talking about cell products, and we have to make those cell products using a laboratory. In order to make a cell product autologous, meaning it comes from the person that's going to go back in, a process called leukapheresis, a wonderful little process that is actually very easy to do and very automated and in very real time, can hook a person up to a machine. They'd pull blood out of one arm, spin it through a very clean and very high-tech centrifuge, basically skim off the level of cells that you want. You can take white blood cells, red blood cells, platelets, or plasma, because the centrifuge basically separates the blood into its component parts simply by spinning it around. And you can specifically take out the lymphocytes out of that, and what happens to the rest of the blood? It goes right back into the person that it came out of. So it's a very efficient way of taking out of the blood just what you want. Cell activation has to be done oftentimes in order to make the cells more permissible or more responsive to the viral vector transduction, because activated cells usually respond better to viral transduction. Transduction is the process that the name that we use to talk about introducing the gene therapy into the activated lymphocytes, of actually that process of actually putting the genes with a viral vector inside the cell of interest. Expansion, we make a certain number of cells, but in order for us to really put them back in, they need to be increased in number and in vitality. And the other thing is that this is a process that cannot always be done in a fashion where you can finish all this done in a period of time to put it right back into the person. And the other thing that's important is that after a product like this is made, a cellular product is made, it has to go through a certain amount of testing to make sure that it is infection-free and other kind of problematic manufacturing problems free. So it is cryopreserved. It's frozen in a very specific way in order for those tests to be done and then become able to be infused into a person. I showed this little cartoon basically of how a person living with HIV is given a medication called GCSF. It's a medication that stimulates the bone marrow to make more new cells. That causes the stem cells, for example, to flood out of the bone marrow into the blood and peripheral circulation. When the stem cells are in the peripheral circulation, we can use leukapheresis basically to be able to pull regular blood through the machine, take out just the stem cells we want, put the rest back, and then capture those cells by a special machine called a Clinimax in order to be able to pull just the cells of interest that we want And then after we isolate the stem cells, for example, the CD34 cells, they are then transduced with the lentiviral vector in order to put the payload of new genes we want in these cells. The cells are harvested, and then a small amount of medication, usually called cyclophosphamide, is given to the person just before they get the cells. The cyclophosphamide is given in order to make space in the bone marrow. In order to put new stem cells into a bone marrow, you've got to have some space for those cells to be able to go into, because that's a very crowded and very active place to be. And if we didn't give those cells space, they probably would not have as nearly a good long-term lifespan unless we made a little bit of space in with the cyclophosphamide. And then hopefully the cells, if everything is done very well, are infused back into the patient, and this often as will be a one, maybe in one case 2-time treatment in order to give new immunity to a person like this. This manufacturing process actually has the days that it takes. The process begins with leukapheresis. This is 1 day, goes right into manufacturing. Manufacturing usually takes around 20 days. Then the cells are cryopreserved. They're held for about 28 days in order to go through testing to make sure that everything is clean and meets, of course, a GMP product specifications. The product is released for human infusion. The conditioning therapy is given, and then somewhere within about 3 to maybe 5 days, sometimes shorter, the cells are infused. So this is kind of the process as we go along in order to go from leukapheresis of taking the cells out, changing them, conditioning the bone marrow, putting the cells back in. So leukapheresis, here's what it looks like. If any of you have ever given blood, leukapheresis is very much like giving blood, except for having 2 lines, one in both arms as opposed to just 1 line. It usually takes anywhere from about 3 to maybe at most 4 hours. It's painless, except for the little tiny nick you get when you have a catheter put into your arm. Most patients watch TV or a movie or relax during this period of time, and it's actually something very, very simple. I was able to help pioneer this in a clinical research facility in the community in Washington, D.C. We apheresed about 40 patients of ours in order to gain cells for research, and we got no complaints or no adverse events. And then the CliniMax Progeny machine is something that's really pretty cool, because this CliniMax Progeny machine from Miltenyi Biotech has been able to automate many of the steps that used to be done separately in terms of cell purification, stimulating the cells, the transduction, growth, harvesting, all in a closed tubular system. So instead of moving even within a GMP laboratory from place to place to place, this is all done in a way that, number 1, is automated, number 2, is all kept very clean because it's inside the same sterile tube system and can really make cell processing faster, cleaner, and actually work, I think, much better for a larger volume sort of way of doing this. The infusion occurs, again, about 3 to 5 days after a mild conditioning, and this is an important part of the whole process. We are, in fact, doing what's called a mini stem cell transplant. And why do we call this a mini stem cell transplant when we do this? Because a real stem cell transplant does not use mild conditioning. They use what's called fully myeloablative. Because when one is treating cancer, like lymphoma, leukemia, we want to blast out the entire immune system of that person and all the cancer cells that are part of that immune system at that point, clean the slate basically, and put in a whole new immune system. That's how stem cell transplants work for persons who have cancer. You cannot have any of those old cancer cells lingering around if you're going to cure the person. So cancer chemotherapy for allogeneic, or sometimes even for allogeneic transplants is a very serious thing when you wipe their cells out, because they're not just trying to create space, they're trying to treat a disease of cancer in the bone marrow at the same time. This is something different. We're not trying to eradicate all the cells in the bone marrow, just a certain small amount of them, maybe 10, 20%, with the low doses of mild conditioning, in this case cyclophosphamide. Busulfan can be used also at low doses as well. So these are different borrowings from cancer transplant that have been pioneered to be used here in order to be able to put those cells into the bone marrow and have them then start doing what they need to do to create more cells. For those of you who aren't familiar with stem cells, hematopoietic stem cells, these are the cells that are very, very special in that they basically are the grandparent cells that can then form every cell that is in our blood. And when we transform and make those cells HIV resistant and protected, every cell that comes from that parent cell is HIV protected for life. And so that's the beauty of what gene therapy can do, is that if we can put HIV protective genes in these stem cells will produce HIV protected cells for the rest of their lives. And as we understand stem cells, hematopoietic stem cells specifically, they are pluripotent. They can go in many different directions, and they never die. They are self-renewing for their entire life. As long as we're alive, they're alive and producing more cells. So it's really getting to the beginning of where the immune system starts, that we are endeavoring to put the HIV protection in those cells and then keep it there for the rest of that person's life. So some important considerations in gene-modified therapy. You keep jumping on me. First of all, can we obtain enough cells during the apheresis? And this is something that, again, sometimes initially was a problem because we were not always using persons who had high enough T cells to begin with. Right now, we know that the best persons who are good candidates for doing apheresis are those that have high T cells over 350 to begin with. But also, when we use G-CSF and other bone marrow stimulating medications like plerixafor, we can get lots of cells. So getting the cells is not so much of a problem as it used to be. Part of it is selecting the right patient who's going to be the person giving the cells, but also it's how we treat them with the medications that stimulate the cells from the bone marrow. And then whether we use mitogen or antigen activation, we know that mitogen activation creates a bulk T cell product with immune properties in order to stimulate the cells in the direction we want them to go, while antigen stimulation would expand cells specific for HIV. So using the antigen, maybe the thing we also want to do is we want to train those cells from the very earliest time as they're being generated to have anti-HIV activity. Because what we're really trying to do here is build a new immune system with new and different and workable HIV activity that it didn't have before. The automation process is something that I think is coming more and more. In the study I did, first of all of the cell processing was done manually, and it was very intensive, and there were potential problems with breakage in sterility because the process was step by step by step, not always connected. And so automated processing is probably going to become more and more common. It's faster, it's more easy to control, it's cleaner, has less problems with breakage in terms of having the cells exposed to something that you may not want to have exposed to in a laboratory. And the technicians sure like it a hell of a lot because they don't have to touch the cells many times. And the other question is about how much conditioning do we need to give? We're borrowing from a part of medicine that has used fully myeloablative, wiped out the bone marrow because they have to, because they're trying to get rid of cancer. We're not trying to do that here. We're trying to just create space in the bone marrow to have the new cells that we're putting back in have a nice home to go back to. So how much Cytoxan or busulfan is needed, it's still something that is under study, how much we need it, whether or not we need to continue it afterwards or not in order to keep those cells stimulated or not, or finding really good ways to have those stem cells home back to the bone marrow after we put them back in, or some of the challenges of how we can improve cell therapy for HIV. I'm going to just kind of run through some of the early studies with HIV gene therapy that dates back as far as 1998. This was with a retroviral vector for RVM for Rev M10 being put into CD4 positive T cells, small number of patients. The treatment's good news was that the treated cells survived longer than the control cells, up to 6 months compared to 3 weeks, done at the NIH and University of Pennsylvania. You'll hear that over and over again. Another retroviral vector, CAR T, phase 1 study for HIV, which modified both CD4 and CD8 T cells with a CD4 zeta, which bound to the GP120 outside on HIV positive cells and caused them to die through cytotoxicity. Treated 24 patients. The transduced cells were only at about 0.1% at 1 year, which means they stuck around, but at a very, very low level. Probably not high enough to really do something effectively, but at least they stuck around. And we know the cells were, in fact, still identifiable 1 year later. And then another retroviral vector in CAR T was done, another study at the NIH and University of Pennsylvania in 2002. And CD4 cell counts increased and trended to a few new patients with recurrent viremia. So these were, I think, some of the earliest studies done with retroviral, not lentiviral vectors, not the best vector, but they did show small bits of interesting information. 2006 was one of the first lentiviral deliveries in which an envelope antisense RNA to CD4 positive T cells were given to 5 patients in a phase 1 study. The cells were well tolerated, the viral loads were stable, except for a decline in 1 patient. There was no evidence of insertional mutagenesis after up to 3 years of observation by Verixis in Gaithersburg, Maryland. This was an important study because of the fact that it showed that lentiviral vectors, which are derived viruses that are derived from HIV specifically, but really clipped down to have only no harmful HIV portions in it, but just the good parts of what we want to be able to insert those genes into the cells of interest. And this actually showed long-term effects and also no mutagenesis, no bad things happening from the vector inserting itself into the human cells. 2010, another lentiviral vector with into this time not T cells, but CD34 hematopoietic progenitor cells, stem cells in other words, in 4 participants who were requiring bone marrow engraftments for lymphoma. This was very much kind of like what was happening in the London patient a few years before, but it had actually 3 anti-HIV strategies: a TATREV, a short hairpin RNA knockdown, a TAR decoy, which is TAR is a gene activator that is on HIV genome, and a CCR5 ribozyme, a way to try to be able to downregulate CCR5. So this was sort of a triple way of being able to try to perturb HIV infection in patients who are also being treated and primarily for lymphoma. This was done at City of Hope and in Duarte, California. Later, the CD4 zeta study that we talked about before had a long found that in 98% of the samples tested, 11 years after the patients got the cells to begin with, that they could still detect the modified cells in those persons, which again tells us that this is a lifelong or at least long-term effect that is happening here. Also showed no cellular abnormalities, no clonal expansion, and they were not immortalized. That's an important term, immortalization, because what that usually means, the cells may have become cancerous. Cancer cells are oftentimes determined as immortal cells because they don't stop reproducing like normal cells do. And so all the fact that these cells did not turn into that, they did not have clonal expansion, was good news 11 years after they were given. Again, a lentiviral vector for envelope antisense RNA in C cells, 17 participants with viremia who received a total of 87 infusions up to a 5-week half-life in the blood and were followed for up to 5 years. Again, the Verixis study that showed 5-year follow-up was actually showing good things in terms of continued detection of the transduced cells and no immortalization or clonal expansion, and some transient changes in viremia. One of the things that's been tough in gene therapy is that what's the best marker to really show that the gene therapy is working? And because we know that changing an immune system does not happen overnight. It happens as a process, because lymphocytes are cells that actually live many years, as opposed to some white blood cells that only live a few hours. So expecting viremia to go down immediately like it does with an antiretroviral medication, a small molecule, is probably not going to happen. It's probably going to go down over time as the immune system builds up. And so this is where I think we really get back to the idea of HIV control, or what we now call more commonly a functional cure. When we really step back and look at it, modern medicine does not really cure many infections. Modern medicine does not really cure many infections at all, not even hepatitis C. What it does is it brings the level of the infection down to a level that our immune system can handle it. So this functional cure would really be the same thing, that we would bring the amount of virus down to a level that our immune system, now that it's been regenerated with cells that are protected from HIV, can handle it. And that's the way we have historically always controlled infectious diseases, either by vaccination, by boosting the immune system to be ready for the infection when it comes along, or antibiotics. You know, antibiotics don't clear out every bacteria in the body. They just bring them down to a point where our immune system can knock out the rest. So this is very much in keeping with how we have treated infectious diseases all along. A sterilizing cure that probably occurred in the bone marrow transplant patients probably occurred because they got massive doses of chemotherapy that killed pretty much every immune cell in their body. But that was the option. That was what they wanted to do, because those immune cells were full of cancer. And then finally, an adenovirus vector delivery of an anti-CCR5 zinc finger nuclease, the Sangamo studies that were done, that actually were treating not stem cells, but again, CD4 positive cells. They actually showed some very interesting initial results here. They used 10 billion autologous modified CD4 positive T cells but only about 11 to 28% of them were actually had the CCR5 gene knocked out, and had a half-life of about 48 weeks, so they were durable. They lasted over a long period of time. There was some protection of the monocyte cells after treatment interruption, meaning the virus did not come back right away. It did come back in almost all patients except for one, who now has a low level of viremia several years after being involved in the study. And this individual who had this, one individual, has a special sort of genetic code in that he only has one copy of the CCR5 gene in his body. So his targeting was easier with this sort of knockout, because there was only one gene to knock out as opposed to two. And he may actually done better because of this, because his targets were not as many. And then some ongoing clinical trials here, a longer, more impressive, longer-term study sponsored by Sangamo, being done at City of Hope. This is actually a very interesting study, because they're taking HIV positive individuals without cancer and treating them as if they had cancer, meaning that they are giving them doses of preconditioning of busulfan as high as you would give someone who has cancer. This study is one that is requiring the persons who are involved in it to live on campus for about 2 weeks. And so far, what I've heard about the study, it's not been made public entirely yet, but they've actually got up to the highest dose of busulfan so far, but have not reported any results. The next study here, the one done in France, is actually using the Calamune vector in patients who have primarily lymphoma or leukemia. As far as I know, they have not enrolled anyone yet in this study, but are looking to do so in order to try to use the Calamune dual HIV gene protection vector, but have not done so yet. And then there's other vectors that actually have multiple mechanisms for stopping HIV, such as the knockdown of CCR5 by shRNA, TREM5 alpha, and also a TAR decoy. Again, the AIDS malignancy group is doing this again, but in only patients who have a cancer diagnosis. So again, many of these studies are still limited to individuals who the real thing they're treating is the cancer, but because they're HIV positive, they're also throwing something into the mix to try to make it something that would be good for HIV as well. Some of these other ones here, they're using zinc finger nuclease. I think we know that before. I was involved in the study, the dual anti-HIV gene transfer study with Calamune in Los Angeles. That study, thankfully, which has ended about 6 months ago, is going to be presented at the CROI conference publicly for the first time to actually show the results of that trial. I can tell you that it was a very interesting trial of learning to give chemotherapy to individuals in an outpatient setting, not a hospital, but an outpatient setting, and actually do the infusions in a way that actually was very much doable. That really proved to me and others that this kind of treatment does not have to occur in a hospital. It can occur in a clinic as outpatients. The other Sangamo studies and other CXCR4 and CD4 Zeta studies are either planned or ongoing now. We don't have a lot of data from them, but this is just to kind of give you a flavor for the fact that many different strategies are being tried. They all kind of get back to the same thing now, is that the cells of interest have become more all the time stem cells, not the CD4 cells, because CD4 cells are already adult, terminally differentiated cells, which are only going to last a certain period of time. They'll last several years, but not for the rest of the person's life, which would mean that you would probably have to treat the person over and over again to give them more of those cells. With stem cells, the beauty of that approach is that you give them the stem cells hopefully once. Repopulate their bone marrow with HIV-protected cells, and then they live the rest of their life with a ever-increasing number of HIV-protected cells that protects them from HIV for the rest of their life. So some of the evolution of gene-modified autologous cell therapy for HIV disease. The current approach is cell modification by viral vectors or naked RNA. I talked about knockdown or knockout, CCR5. Introducing other inhibitors of HIV replication or cell infusions after non-myeloablative or non-bone marrow wiping out chemotherapy. Some of the critical events for enabling events, decrease the potential for off-target gene modifications. This simply has not been happening in studies to date to a great extent at all. Target vectors to preferred T-cell subsets. One of the things that, of course, vectors can do is that they can be targeted to certain cells by what kind of molecules are on the outside of those vectors. And so in that way, we can get better and better at being able to have the cell therapy go only into the cells we want it to go in. Creating more, the vectors we've been talking about are actually pretty simple vectors, usually having only one or two, a couple of times 3 different payload genes. But we can actually put genes in there that actually work together and may actually hit the virus in more than one way, or be able to find ways to be able to intensify the survival of the cells over time. So more can be done certainly in vector technology as well. And the last thing, of course, would be to transition from ex vivo to in vivo therapies. Right now, gene therapies with cells basically has to take the cells out of the body, into a laboratory, very cleanly in GMP facility, transduce them, and then put them back in. There is a possibility that we can actually do this in vivo, to actually have the person inject the gene therapy into their blood, have those vectors go right to the cells we want to change, and never have to take their blood out at all. So this is another down the road sort of way of trying to make this process even easier. I think I'm going to skip over some of those things. We've talked about most of those already about different vectors. The last thing I'm going to touch on is kind of views from the community. And I show these next 3 slides basically because when I first started getting involved with this several years ago, around 2006, there was a big concern is, HIV therapy has gotten simple. Many people get started on 1 pill once a day. Many people who've been on therapy for a long time may be on 2, maybe 3 pills once a day. It's much easier than it ever has been before. So why are we trying to cure? Why are we trying to cure? Isn't the HIV problem solved with just small molecules? And when you really go out and start talking to people who are HIV positive, there's lots of reasons why they certainly perceive it's not done. This area of research of human therapy is not done. For one thing, medically, as Bill told you, certainly because we've brought down someone's viral load and had their T cells come back up, they're definitely not out of the woods. We thought they were out of the woods back in 1996 when we could suppress the virus and saw T cells come back up. But over the past 20 years, what we have seen is that there is an increase in other sort of what I call second generation HIV diseases. More cardiovascular disease and in the HIV negative population, more neurologic disease. liver disease if there's hepatitis C viruses involved oftentimes, bone disease from the medications, renal disease from the medications as well. And so these individuals are living, but they're not living in optimal health. Physically not living in optimal health. The other thing is that once someone's taking a pill every day, the stigma that still goes along with HIV, and you might or not understand or know this, but simply saying that you're HIV positive makes people scared of you still, makes people afraid of you. Even though we know, for example, if you take your medications every day and U equals U, undetectable virus in the blood means you can't transmit to anyone, people are still very, very hesitant to interact with people who are HIV positive. So the stigma is still very, very much there. So cure is something that I think lots of people who are HIV positive, especially those who've been living with the disease for a long time, are very interested in. Yes, there are concerns about what it's going to look like. Yes, there are concerns about how much time, energy it's going to occur. And there's been some really great publications from individuals in the HIV advocacy community really looking at some of the words that they would like to have say in here, because using simple things like saying eradication or elimination as opposed to sterilizing. We use the word sterilizing in a medical sense, but the word sterilizing in a non-medical sense has the connotation of meaning you're taking someone's ability to have children away. You're sterilizing them from their ability to have children. So simple words that can be very important in terms of talking to the individuals about HIV cure are really, really important. But I know you're going to see perhaps a couple of videotapes of individuals who actually are confronting this in their daily lives. But this is a very loud voice. This is a very loud voice in terms of where the research and the next step, where the next step in terms of making HIV therapy better really is. Is this scalable? Eventually, certainly if it could be an in vivo treatment, it certainly could be scalable. In order for people to be treated one time, as opposed to many, many years with the $300,000, well, I think it's going to be even higher than that. Because see, right now, a 3-drug regimen costs somewhere between $25,000 to $36,000 a year, and if they live 20 years, it's already over that $360,000. It's double that. So economically, it even makes sense that if we could find something we could treat people once with, and stop their use of medications, their lives would really change, and the health of our public health would change as well. So I think this is something that is still going to be very interesting and doable in the future. And I'm glad that there's companies like AGT exploring this. Thanks very much.
So I'll call my own number now. And did you guys have problems with the auto advance when you're using the computer or when you're using the?
It's just the clicker. But just use the clicker. It works fine. Just, not sure why I was doing that.
So we'd like to introduce you to two persons with HIV that were involved in gene therapy clinical trials. And David Hardy mentioned the one person who had been successfully treated in a study by Sangamo. And that person is now agreed to be an advisor to the company, and I think you'll enjoy meeting them electronically here.
I started taking HIV meds 1 year after my diagnosis in 1989. I've been on just about every HIV medicine there is because once you take the HIV meds, you can develop resistance to them if they're not taken properly. That's what happened to me. I became debilitated with HIV wasting syndrome, which is a syndrome which basically you lose your weight and you become dangerously thin. I had to enroll in clinical trials because the drugs I was taking weren't working. But I would like to see the day where I don't have to take HIV meds anymore. So I'm interested and excited about cure research because isn't that what we're looking for? That's the dream.
I became interested in what they call cure research when someone named Timothy Brown was cured of HIV, and the way they did that was through gene editing. The study that I participated in, they took my cells from my plasma, they reprogrammed the stem cells so that those cells cannot be infected with HIV. And then the part of the study is then you go off your meds and see what happens. I had this unique experience that I'm still off medications. So what that means is that my body naturally controls the virus, and that for me is vastly different than what happened before I went into this study. HIV is so prevalent and so destructive that finding a cure for it would really, really change the whole paradigm of the disease. I don't know if you can calculate the volume of meaning for that. It would be such a wonderful and beautiful thing.
American Gene Technologies is where we need to be with cure research today. So that's why I think we need more people, not only patients that are enrolling in the studies, but we need a global response. We need a global effort and support in ending HIV.
So as you can see, the members of the community and people with direct experience are highly supportive of this approach. And I include these videos and I include these comments because we need to push back against this position that this is a done deal, right? And that no more is needed to work on HIV. I can guarantee you every pharma company involved in this game is working every day to develop newer, better, safer, longer acting drugs. And there are alternatives, and the combined effort is really what's called for. So I'm going to speak very briefly and to try to introduce you to exactly what American Gene Technologies is doing about this and what we learned from the lessons that you heard about in the previous two presentations. So we've developed something that as part of our program towards creating a cure for HIV, and this is the first step for us. It's a cellular product modified ex vivo called AGT-103T. It's a single dose autologous cell therapy, and it'll be delivered to persons with HIV while they're still taking their normal antiretroviral therapy. And our goal and the key difference between us than anything that's come before us is that we are specifically evolving this product to be an advanced immunotherapeutic in addition to just a simple modify T cells and put them back in. And that's because we believe that the key to long-term control of HIV and to eradicating HIV from the body is to regenerate a normal antiviral immune response. So then the immunity will now treat HIV like it treats every other virus and either suppresses it, like you carry cytomegalovirus or other viruses in your body all the time, you suppress them, you have not a problem with them, or you can eliminate it by just eliminating the infected cells. But in order to do that, we don't know of a better way than to use the immune system. So the product rationale. You heard most of these lessons in the previous two presentations. Treatment is simply not a cure. And I think all of us now believe it will never be a cure. And we're very grateful for the benefits of treatment, but it's not going to solve this problem. The key event in HIV infection is, as was mentioned before, is that HIV depletes the very HIV-specific cells that are required to generate antiviral immunity. And so it's feeding off that population. Imagine this vicious cycle. The virus comes in, it activates the CD4 cells by their specificity for the viral protein. That cellular activation feeds the replication cycle of the virus. And this vicious catalytic event occurs until eventually HIV exhausts those cells and then drops down to some lower level of virus that persists in the body. But if you look years and years after onset of therapy, you don't see a recovery of these cells. You would expect at some point that you'd suppress the virus and these helper T cells would come back. It simply doesn't occur. 20, 25, 30 years of treatment, those cells are still at a level that's at least tenfold and sometimes 100-fold below what we recognize as effective for antiviral immunity. So you just don't have enough. And when you don't have enough and you take the medications away, the virus gains a foothold and just plays this game out again and uses those same cells for explosive replication and deletes them again, and you're right back to where you started. So we know what that subset is. It's the HIV-specific subset. But imagine this number right here, 10 to the minus 4 frequency. Really, when we have an effective cell that's protecting us from influenza, it's between 10 to the minus 2 and 10 to the minus 3. 1 in 100 to 1 in 1,000. Here we're 1 in 10,000 and often lower. We just use this number because below that our measurements are really not very good. And so if we can reconstitute these cells, we should be able to make immunity. But if we reconstitute the body with these cells, and they're still susceptible to HIV, it won't make immunity, we'll just feed the infection. So that's where the gene therapy part comes in, and I'll explain that as we go along. So what did we want to do? We wanted to make a potent and cost-effective therapy. Jeff Galvin told you this before, and we'll show you exactly why we've engineered these features into the product. We're going to manufacture it ex vivo. That's the logical, reasonable, and responsible place to start in any of these programs. We have the long-term goals of going in vivo. We have the long-term goals of simplifying this product. But you start in the responsible position and show that it works. So what are the critical components for us? Well, we needed a potent lentivirus vector because we have to be able to modify these cells in a way that doesn't change any of their normal properties. It does not alter their ability to participate in the normal immune response against HIV, but prevents them very effectively from attack by all types of HIV. We needed a scalable and consistent process for doing this repeatedly, and as I said, to do it at a cost that's within the ranges that we thought were correct. And we need to be able to make a lentivirus vector transduction of these cells, but we only want to alter the cells that are responsible for interacting with HIV. So when I call GAG-specific cells, GAG is a part of HIV. The T cells are responding to it, therefore we call them GAG-specific. And those are the ones that we want to modify with our gene therapy, right? You hear a lot, and you'll hear a lot more in the news about people who want to wipe out CCR5. They want to remove it. They just want to put in something in the body that just destroys all CCR5. The problem with that approach is you use CCR5 for many important immunities. You know, some great examples. People who are CCR5 negative will be in the morgue pile, not in the living pile after a pandemic flu. It's absolutely required for that resistance. It's absolutely required for resistance to West Nile virus infection and a whole bunch of other infectious diseases. And many common infectious diseases are modulated by your CCR5 status. In fact, a recent publication came out looking worldwide at everybody who's born CCR5 negative and showed they have 29% reduction in lifespan. This is not free to modify CCR5 everywhere you choose to. So we've targeted this exactly where we need to do it, and we spare the rest of the immune system. And that's another critical breakthrough that AGT has made. So what does the vector look like? It's quite simple vector. This is a standard lentivirus vector. There's a lot of engineering in here that David Hardy mentioned. That's a couple of decades of engineering to make this thing as really extremely as safe as possible. So it can't really ever function as a virus. Just the information in the middle can be picked up and delivered to a target cell. And that information is very simple. It's 3 microRNAs. It's 1 to reduce CCR5 levels, and 2 to attack viral proteins. And it attacks the virus in 2 highly conserved regions. And very importantly, one of those viral proteins is VIF. And if you know a little HIV biology, or maybe more than a little, I guess, VIF has many weird functions in the infection cycle. And one of the things VIF does is it arrests the proliferation of the cell that it's in. It's an anti-cell cycle protein. So a VIF positive cell can sit there and manufacture HIV particles without dividing. And that's not good for us. We want a proliferation of these cells, so one of the targets we knock down is VIF. Okay, so we block this vector, and I'll show you the data really quickly. The data are not as important. Is that we can block the CCR5 tropic virus, which everybody talks about, and you saw cartoons about, and heard about Berlin and London and Dusseldorf, and I guess we do the world tour of patients that are treated this way. But that's not the only thing you have. And David Hardy told you that there were 3 people that looked like they benefited from these hematopoietic stem cell transplants using CCR5 negative donors. What about the other 42? The reason why they failed is because they had traces of this type of virus in the body before the transplant occurred. And as soon as they put the transplant in, that other virus just grew up like that and took no time at all, and it was right back to being a highly lethal infection. So we want to be able to block both. And we want to then, the third thing is, if you're just engineering CCR5, if that's all you're doing is to knock down or knock out CCR5, what about the cell that's already infected, right? That cell's still sitting there, and that's the reservoir cell. It's capable of releasing virus when it's reactivated. So this type of vector not only blocks the majority of viruses on their way in, it can also block a virus on its way out. And so we get very effective suppression of those. Now the last thing is, all these things, right, keep focusing around the GAG-specific cell. And I told you that GAG-specific cell is the preferred target of HIV. So guess where the latent proviruses are that can come back? They're also in the GAG-specific cells. And if you can develop a process, thank you very much, we've done that, that can efficiently target those cells to a very high level, you've not only protected in the future against HIV replication, but you've dampened the source of this rebound virus that can come back after treatment interruption, right? So just to give you a little bit of data, I won't show you these things. If somebody really is dying to look at flow plots, I'll stop and do it. But there are a few things that we want to overcome in our testing of the vectors. So this is just to tell you that we actually do things in our lab. And so first, you want to show that something like your CCR5 knockdown is incredibly specific and doesn't affect anything else other than CCR5. So we did an exhaustive test for specificity, and we convinced ourselves that there really is no other affected target other than CCR5. The microRNAs against HIV, we screened every genomic database on the planet for every organism. That includes bacteria, plants, animals, I don't know, whatever else, lots of other viruses. The only targets are in HIV. The second thing is we show that when we block CCR5, it's no mystery to then convince you that we block the infection by CCR5 using viruses. And the blockade here is greater than 95%, right? And that's good because when you do these in vitro models, you use very sensitive cells and you use very high concentrations of virus because you don't want to wait for a long period of time to get the result. 95% is about as good as it gets in this type of study. But we get 92% blocking the CXCR4 tropic virus. So we've accomplished that. So why not just get rid of CXCR4 on the cell? Well, then you have a CD4 cell that has no function. They require CXCR4 to get in the lymph node and to move around inside the lymph node to execute their normal biological function. So R5, that's a receptor that's used out in the periphery for T cells to go into tissues. X4, off limits. You can't take it away. So our vector gives you very good protection against those two types of virus. And lastly, this is the reservoir assay. You take a latently infected cell, and we know when we treat that cell, it'll make gobs of HIV, and here we've reduced it by a very large amount. I think this number is close to 90% reduction as well. Okay, so here's our now well-known friend, the leukapheresis machine and the Prodigy machine. And then what happens after that now is quite different. So many of these processes are becoming common. They're being adopted for like routine CAR T manufacturing and routine other types of cell products. But our process is quite different in a few very key steps. So number one, we don't use random or mitogenic stimulation of the CD4 cells. We use antigen stimulation. So we use small synthetic peptides that represent the GAG gene. So we're stimulating those cells that are present in 1 in 10,000 to 1 in 100,000 of the CD4. Okay? Very rare stimulation. The next thing is that so we leave all the cells together in a peripheral blood mononuclear cell mush, because that's the best way to get an antigen response. But after that, we don't need everything else. We just need the CD4 cells. So the next thing we do is we take away cells that are non-target cells. We drastically reduce the number of cells that are in the process. When you drastically reduce the number of cells, you can drastically reduce the amount of vector you put in. So compared to something like a CAR T process, we may be operating with 1/7 to 1/5 the amount of vector that those guys are using. And if you know CAR T, vector is the driving cost in the manufacturing. Okay? So getting the vector number down is absolutely critical and getting it down right away. But this does more than that. What happens is the specific activation of those GAG responsive cells means we only transduce them. And about 75 to 80% of the transduction is only occurring in those GAG specific cells. There's very little off-target event. So this is the most focused, the most surgically precise gene therapy that's ever been made of this type. Really only going after the cells that we want to target and sparing everything else so we don't introduce immunity. We have like a little joke inside the company, you know, you don't want to treat one immunodeficiency disease by giving you another immunodeficiency disease. So we try to focus this as carefully as possible. Then we learned some other lessons here. We learned that the existing automated processes didn't have the volume or the culture requirements for these very special kind of antigen-specific cells. So we modified this process. We went into some what's called static culture system. But then after that, it's purify the cells, wash them, put them in cryopreservation, and begin your sterility testing before conditioning and infusion. And here's the real takeaway message. In the incoming material, we have about 1 million of these GAG-specific cells. In the product, we have about 1 billion. And they are about 85% transduced, modified genetically. So this is really quite a spectacular product. So these cells are almost equivalent to the number of GAG helper cells in the entire body, and we just started with a small sample of blood. To give you a look at what the process looks like, I realize this is somewhat unreadable down here, so I'll read it to you. The bottom line is just the total number of GAG-specific CD4 T cells in a product. And it goes from 1 × 10⁹ to about 3 × 10⁸. And our original target product profile called for achieving 1 × 10⁸. So we blew way past that a long time ago. And in the world of antigen-specific enriched cell therapy, you really start seeing very high efficacy across the board in cancer, in infectious disease, and a number of other areas when you hit about 10 or 15% enrichment. And we've got 10 or 15% enrichment every day. We go up to about 25, 27% enrichment. So imagine starting with a cell that's 1 in 10,000, and now it's 25% of the culture. That's a pretty spectacular achievement. And out of all these processes, there was one failure, and that was due to an oddball situation in the person, nothing to do with the technology. And so this is a robust, reproducible process. Okay, this is a slide that just flexing some flow cytometry muscle to show you that there's really a very high percentage of transduction in the GAG-specific cells. And that's what we were after all along. So that's really, the components of the product are all borne out, and they all obeyed what we really tried to do before. So I just summarized this process, right? So it's very reproducible, as I just showed you across, and those runs that I showed you, those cell processing runs, were done by 2 different manufacturers. And we had some people in there, some donors, who came back for the second manufacturer and got almost exactly the same results again. So we're not plagued by the current issue which is plaguing CAR T, which is the inability to manufacture products. Some of those problems in CAR T are due to the fact that you're collecting cells to begin with from very sick children, right? We're collecting them for healthy people on ART, and we get a very robust process that's reproducible. It's scalable. I mean, I don't know how many Prodigy machines Miltenyi can make every year, but all you need to do is just have a whole farm of them, and they run. And when you have these machines, you get a big cost savings because the labor hours per product goes way down. And it's absolutely reproducible, same software on every machine. And there's 20 manufacturers trying to make competing products right now. So soon there'll be a lot of choices in this area. And we think that we've made it economically feasible that, that by controlling the cost of goods and controlling the manufacturing time, that we can get this, reduce the price of the product. So we showed you Matt Chappelle, the second person that you saw in that video. They're both named Matt, which is kind of interesting, and they both were friends, and they both signed up for the Sangamo study together. And the first person, Matt S., was talked Matt Chappelle into going to the study. And then Matt Chappelle eventually got durable virus cure, and Matt S. is still looking for his breakthrough. But both of them are very enthusiastic. So that Sangamo study is the one and only successful gene therapy study that we have for one person, right? But if we look at Sangamo overall, what you'd find, and this is a theme through the recent gene therapy studies, is people tell you they feel better. They just plain feel better. And it's not that they just report this anecdotally. It's that they have less clinic visits, they have less supportive therapies, they have less, imaging things to find out what's going on in your kidney. All this kind of stuff is reduced. So their burden on the healthcare system certainly went down, and they just feel better all the time despite having to still take ART. So we think we're really the first gene and cell immunotherapy. We think that we're the ones that really built this into it as a major objective. And if you look at the Sangamo product and ask how many GAG-specific CD4 cells did they modify to make CCR5 negative compared to what we have in our dose, and there's a 2,000-fold difference. So if the hypothesis is correct that this is what you really need to fight HIV, the product delivers, right? We're doing this for people with chronic disease. We have tested this manufacturing for people in very late stage disease with CD4 less than 200, and for people in salvage therapy, two groups that are in dire need of help in HIV. And we can make products from all those individuals as well with a few little tweaks. And a point that Jeff introduced you to earlier, that we want to immediately reduce the requirement for antiretroviral therapy. But we think that by starting a long-term reconstitution of the immune system, everything just gets better and better. And there's nothing else that's on duty 24 hours a day, 7 days a week for the rest of your life. So where are we right now in this whole business? So this is the timeline for the AGT-103T product. We've submitted the IND. We're cleaning up the last few issues in the CMC area that were raised by the FDA. And these are primarily issues that have to do with record keeping by manufacturer and things like that. And so we have no issues related to the protocol and no fundamental issues related to the product at all. And but every time you put something this new in front of the FDA, they're going to all of a sudden think of new things to do. So we're complying, and we're being good, obedient people to the Grand Master.
David, just one quick question. Is that related to the master file? Those questions?
I don't want to get into those kind of details, John. So, because we don't hold the drug master file. So that's I'm talking about somebody else's stuff. So trial initiation, we think will be February or March. The first patients will start clinical manufacturing, and it'll take about a year to do the phase 1, and then we end up in the long-term follow-up. Our real objective through all of this phase 1 study is to gather objective markers of changes in immunity in these individuals, to be able to either stratify the group to learn about responders versus non-responders, or to provide a better justification for doing a treatment interruption study that will then really measure the efficacy. Now, AGT, we have busy people that don't want to sit on their hands. We're remaking these vectors. We have a very potent version 2 coming. We are re-looking at the cell process. We have been experimenting with many changes to the cell process. And we are looking at all kinds of modifications in our lab scale in order to be able to have a second gen if needed. Okay? The clinical trial looks basically like this. It's in 2 parts, and cleverly named Part A and Part B. And so in Part A, it's just the safety of the cell product alone. The FDA has asked us to stagger this since it's a very new thing. So we have 3 people that we'll treat, If they're okay, we'll treat another 3 in the group 1. And then in the second part, in part B, is something we thought about quite a long time ago, which is some people may have just a little bit less of these CD4 GAG-specific cells than you'd like to start the whole product. And so we figured if we take an existing so-called therapeutic vaccine that's already been tested in HIV-positive patients and known to stimulate the CD4 T cell response, we can immunize with that and see if that helps improve the product. And so that's a randomized 1-to-1 study, and that'll complete phase 1. As I mentioned, I gave you the status of this already. We have sites selected. We've already vetted everything. We've completed a long specimen collection study, which is kind of a good way to get material to do the cell process development, and also get you engaged with the sites and get them warmed up a little bit. So we're feeling really good about all these things. Just before I quit, I'll just introduce you to the team. You met Jeff before. You unfortunately had to listen to me. These are the other major members of the team there. This is really a homegrown product, right? This is 100% technology from within AGT. The product's completely developed in AGT, and all the future generations, of course, will. And we just had another patent issued on the 3rd of December, so I think that's the 4th HIV patent, and we're moving along with that stuff. And with that, I'll stop. And what I'd like to do now is invite any questions you have for anyone. And Jeff, if you want to chair it formally, or if you want to just let it happen, however you'd like to do this.
No, let's just let it happen. Did anybody have any questions for any of our experts or for me from the presentations?
Yeah, so quickly, you mentioned before, you basically simulated a CAR T process in order to treat for HIV. But one of the comments has been in the CAR T, it's the cost of treatment per patient. Have you done some econometric model, what you foresee as being cost per patient? Now, I do hear that you don't need that much sampling, you only have to do that. And you say it's cheaper, but you haven't given me any numbers as to what % you think is going to be.
Okay, so the question is, just for the people that are watching this on the video, is that since our process parallels CAR T, the question was about how does it compare economically to CAR T? What will the cost be of a treatment using this? Okay, so my expectation is that this will retail somewhere between $300,000 and $500,000 per patient. We think this will be justified because of the long-term cost of treating the disease, and also all of the collateral costs to society in the cost of productivity losses and so forth that HIV-infected individuals are currently experiencing. So we think that there would be little resistance below the 500,000 mark. At that point, if a pharma company were to bring this out using our current technology, they might be getting about an 80% gross margin at that rate. And that's sort of what we're looking to achieve. Now, as David was mentioning, this is our first product. And it is our philosophy to try to obsolete ourselves so that somebody else doesn't do it for us. And of course, driving down that cost is really important. David has already found some angles on this where we may be able to reduce the cost by a factor of 2 in the near term, and perhaps up to 10 within a half decade or so. So I think we can, over the long term, bring the cost down of the treatment. I think we can pass this benefit to the public and to the patients, because one of the important things that's different about gene and cell therapy is you don't have to think about each one of your siloed developments as being critical in your revenue stream. It's when we go out there and sell a drug, it's more like we're going out there again and selling software. There's a profit maximization point that doesn't have to be at the point of pain of your most wealthy consumers, right? So we don't have to maintain a high price on this product. If you can spread it out to 37 million people, right, there is another pricing strategy that would profit maximize And the company itself is not dependent on this as its only revenue source, because we're going to take our platform, that $2.4 billion worth of IP, and we're going to do drug after drug after drug. Now, this is a very interesting thing. Talking about platforms now, they haven't been that important in previous drug development modalities. They're generally a more efficient way to do drug discovery, which is really still somewhat a random event. You generate 10,000 molecules and you screen it down to 2 that are worth testing in a mouse. It takes you 2 years and $10 million to get to those 1 or 2 candidates that are worth testing in a mouse. If you're lucky, it makes it into the clinic. If you're exceedingly lucky, it makes it out to the public. That's a very expensive process to go through. We figure, and remember I told you the idea of efficiency in this business would be a critical differentiation factor between AGT and other companies. We figure that over the long term, from idea to first human efficacy could be brought into the $10 million and 2-year range. Think about that, with a lot of go, no-go opportunities along the way. Imagine that at about $100,000, we would have roughly 80% confidence that it would work in the clinic or not. And we could pull the plug on the project at that point. That's because we can simulate a lot of the clinical experience in cell models. This is a deterministic science where we can go ahead and put things into the vector that are on-off switches that can look inside the cell that this virus infects and determine whether the cell has a particular disease or is of a particular cell type. So we're able to predictively narrow the treatment down to the targeted tissue, and this avoids a lot of the side effects. And so, and that can be simulated in cell models where we can test it in cells that it's supposed to turn on in and ones that it's not supposed to turn on in. And we can get a lot of data before we get into that very expensive clinical phase. So, so we are going to, I think, disrupt people's thinking about platforms. Platforms are going to be more critical in this new drug development modality. Now, let's talk about one platform that you heard about today. Nobody said platform, but what's the difference between HIV and hepatitis B? The T cell that treats it, right? So all of those patents that our scientific team have put together, that are this whole process of isolating a particular T cell and enhancing it in a way where it can go into the body and it can better maintain the suppression of viremia than you can naturally, right? And all of that applies to every chronic viral infection. So we won't be thinking about how do we ratchet up the price of our products to the audience's point of pain because we hold the only solution for an audience, and as a corporation, it's our responsibility to profit maximize, right? We won't have to slowly ratchet up that price to their point of pain of a hostage audience. We can go on and take a big chunk of that project and redeploy it over here and here and here and here. What does that sound like again? It's like the computer industry. When I was at Apple and we sold the first Mac, which did almost nothing, for $2,000, and a bunch of people bought it, we weren't thinking, wow, they can't live without it. Let's sell it for $2,100 next year. No, we were thinking, how do we drive the cost down so more people can buy them? And how do we take this platform and blow it out to so many applications that pretty soon everybody has to have one of these in their house? They can't live without it because of the convenience factor. You sell computers because the people that buy them are making an investment in something themselves. I think long term, this is going to be a total change in the thinking of pharmaceutical companies. They won't be able to survive without rearranging their thinking. You're going to have to make every drug an investment in the consumer. They're going to have to buy something that gives them a return, right? A return in quality of life, a return in longevity. Drugs that give you 6 weeks of extra overall survival for $125,000 aren't going to be able to sell in that environment. If you can knock back cancer for 5 years and you charge $300,000 for that, you've left $200,000 on the table for the consumer, society, whatever, right? That could be the new economics of this. Now, that's a very long answer to your question, but I think that the economics is a flowing situation. It is going to change over time as we refine our process of developing new drug candidates. I think we have a intention. I think we have a reasonable probability and methodology that seems effective in creating new economic opportunities that will better match solutions to patients and provide economic incentives for insurance companies and society to get behind those solutions and wipe out diseases like HIV, but also many other diseases. And we are seeing this in our data. So we just got a question about what would be the effects of completely eliminating CCR5 in the body, and what would be the effects of eliminating CXCR4? Because these are two entry points or important molecules on T cells that allow HIV to bind and invade the cell. David?
Well, first of all, we get some kind of information about this from the development of drugs that block those co-receptors. And if you want to hear more about that, maybe David Hardy could say something. Because CXCR4 blocking agents are incredibly toxic and incredibly dangerous, and were abandoned early on during clinical development. With all kinds of very unexpected toxicologies. Some CCR5 blocking agents also have the same problem. They had cardiac toxicity, but they were able to be managed by proper dosing and formulation. We think that CCR5 is very important for a very wide range of immune control over infectious agents. So when T cells leave the blood and want to go into a tissue to attack a site of viral infection in the periphery, let's say, that's CCR5 dependent. So we don't want to knock that out in general. In HIV, we're fighting the battle inside the lymph node. And in the lymph node, CXCR4 is critical. CCR5 is a little bit disposable. So when you fight the battle in the lymph node, CCR5 is okay. When you have another T cell that has to go out and do something distally, no, not okay. And that's why we prefer don't touch the CXCR4 or else you'll disrupt lymph node function. Keep your CCR5 modification to an absolute minimum so you don't eliminate it anywhere except where you're required to eliminate it for the product.
Questions? Yes.
As you await the IND approval, you mentioned the FDA questioning about records, and then as questions arise about gene therapy, how does that play a role in receiving approval, or has the FDA essentially said that aspect of it is okay?
Oh, no. So the product.
Repeat the question.
So the question was, are the FDA's concerns about the product details about this specific product, or are they broader about gene therapy? Hope I captured the question correctly. I think that the issues about gene therapy broadly are really just about gone. And you see during Commissioner Gottlieb's term as head of the FDA, he really put in a huge number of changes and new policies to facilitate the review of gene therapy protocols, to put more staff into that part of the FDA to deal with this. I think they are welcoming this because I think that they see the same way that most of us do, which is that the kind of toxicities and other types of issues that you need to deal with are routine. They're not drastically different one product from another. They're variations on a theme, but you have this core technology where the bits and pieces are all the same, and you don't spend a lot of time worrying about that part. The issues that came up for us are small things, like how was this recorded, right? Where did you put that during that time? What was exactly on the label? These were just these kind of things, and you don't know exactly what the agency's preferences are for this until you submit the whole document to them and get that feedback. I think this is absolutely normal for these kind of products because they're breaking ground. You know, if you're making a small molecule, you go to a toxicology company and they say, do you want menu A, B, or C for developing your product? And then you just pay them the amount of money and a big ream of data comes back and you stick it in the IND. There's nothing like that for gene therapy yet. That's still evolving, so there's a lot of customization, let's say, that has to be done.
Yes.
Wondering what type of challenges you've encountered or anticipate encountering regarding capital raising or courting partners for commercialization.
So the question is, do we anticipate any problems continuing to raise money for this, or finding partners for this development, or creating a long-term sustainable company? So I'll say that I'm exceedingly optimistic that we'll get this thing to the finish line, and that we will implement our broad vision for the company. And the reason is that even though we have no lead biotech-focused VC, we've raised nearly $40 million through our own capital desk. We did outreach to the same sort of people that put money into VCs, and what we ended up getting was not just the same kinds of money, but we got money that didn't have the same tight time frames that focused VCs normally have. They have to look at getting a return in a fund in order to raise the next fund. And so a lot of VCs have sort of a 3 to 5 year time frame. Biotech VCs maybe a little bit longer, but it becomes critically important to become very focused and get to some sort of liquidity point. Well, we remained free to go where the science took us. And that was the only way to develop this broad platform. We wouldn't be sitting around with $2.4 billion worth of IP on less than $40 million of investment. Not bad. Nor would we have 3 products in our pipeline of HIV, phenylketonuria, and immuno-oncology. We probably would be a phenylketonuria company. Why would that be? Monogenic diseases are just believable out in the layman public, which I would include the biotech-focused VCs. They would say, that's your best bet, and do it with AAV. And so we have really broken free from so many of what I consider to be stigmas and prejudices in the gene and cell therapy space, to look at the broad picture and really find low-hanging fruit, brass rings that we could grab. Nobody would have had the patience for 11 years of development, but now I think we have an opportunity to come out of the water in a very strong way. And my confidence level in showing human efficacy in this project is quite high. And you can't take that to the bank, but at the same time, I think a lot of the top scientists in the country agree with me that we've got a really good shot on this one. And so I think my confidence doesn't come from nowhere. Now, what happens if we show first human efficacy next year? Let's say that a third of the patients are durably in remission. Changes the mindset of the patient population. Now they have an expectation of cure. What does that mean? That puts tremendous pressure on the industry to get on board. As a matter of fact, Gilead's in a very uncomfortable position, right? If the cure works out and they don't get it, they're the ones that are terminal, right? There's another interesting aspect of this. Remember, people have to be well controlled on antiretroviral therapy before they can take a cure. They have to have an immune system before we can protect the immune system. What does that take? 1 to 3 years on antiretroviral therapy. How many people in the US are on antiretroviral therapy? There's 1.1 million people infected. It's somewhere south of 300,000 that are actually on antiretrovirals. That means that the antiretroviral business could go up substantially if people weren't faced with those severe toxicities of taking that daily pill. That's why a lot of people avoid it, right? And it's a lifetime commitment once you get on that pill. So what will happen is Gilead's business would go up overnight. All they have to do is announce, hey, we just purchased the cure. It's credible. You read about it. Several patients are in durable remission right now. We're going to develop this. It'll take a few years. You should get on antiretrovirals. Where are you going to go if you're an HIV-infected individual and you realize, hey, now it's just 1 to 3 years before I can be cured? You're going to go to the brand that's selling the cure, no one else. They could enjoy an $8 billion pop in their revenue on an existing drug first year, never even putting out something new. In the meantime, they'd be looking ahead to potentially $10 billion a year in cure revenue. So you're talking about Gilead being able to regain the story that they had with a longer time frame, with boatloads of IP protection around this. This is really unique. For decades, this is what they're looking for. That's the positive side. What if J&J buys it? Gilead will have to tell the story to a room of analysts that sounds more like this. We don't expect our franchise to go away overnight. Right? That isn't going to make you go, buy. Right? So I think that, it all is contingent on first human efficacy. I think if we get a handful of patients on this very inexpensive therapy, very commercializable, very practical. This is not the $500,000 worth of viral vector that Sangamo had to use in their study for each patient. This is $30,000 worth of vector. There's other expenses around it, but this is tremendously practical. So if it works, if it gets a reasonable percentage of the patients, we're on our way, and I think Gilead has no choice but to partner with us. Or J&J will do it. It's an opportunity for somebody to either maintain, continue to be Gilead, or for somebody else to become the new Gilead. It could even be us. It could be with one of your banks. Why not do an IPO? Maybe if we sell it to Gilead, we're not maximizing our opportunity in there. So there's a lot of ways to slice and dice this. We've raised more money in this last quarter than we ever had. We've had successively higher valuations, starting at $4 million in 2013 to pre-money, where we raised $1 million. This year we're doing a $25 million raise at a $240 million pre, and it's coming in faster than ever. It's a great investment. I think that the potential on the other side, bring back a handful of cured patients, all right, that's a $15 billion value to Gilead. What would you pay if you were a $70 billion market cap company and you could get back to $130 billion one direction or down to $35 billion the other direction if J&J started to steal your ART revenue? How much would that be worth to save that swing? We're talking about a $90 billion, $95 billion swing. So I think it's $15 billion. That is, think about that. That's 60x the current market cap of our company. So there's a lot of opportunity in there for an IPO, for more high net worth individuals, more family funds, more boutique VCs or general VCs to hop on board. There's a really good upside. What about the downside? What if we were to totally fail? I don't think that that's even in the realm of possibility without a nuclear war or a complete financial meltdown. Okay, well, after, we clean up the ashes of the nuclear war, there's still IP. So the IP itself has value. Granted patents, published patents, data, and 28 patent families. Just one patent family is a unique approach to immuno-oncology that is unmatched as far as I can see anywhere else. We can literally stimulate your natural immune system to rise up and chew away epithelial malignancy at 300 to 600 times the normal rate, and we can do it safely. And all the theory and all the experimentation backs that up. I think we're a couple of years away from getting that into the clinic. Our collaborators, the Stanford Medical School, Dean Felsher, Dean of Translational Medicine, and a globally recognized expert in hepatocellular carcinoma. I think we can show this working in liver cancer, the most deadly form of liver cancer, sometime in 2021. But the patents themselves are really exciting. When I tell people about this that understand gamma delta T cells, it's a head slapping moment. I mean, literally, they're like, of course that will work. Why didn't I think of that? Right? Anybody that knows type 2 gamma delta T cells really well and what their function is in the body, the elegance of this, the sophistication of this, the natural safety of this is striking. Okay, so we can sell that out for $500 million or $1 billion, right? We're not going to ever get $2.4 billion for our IP, if we sell it out. But $500 million, yeah. So what does that mean? When people are investing at a $240 million pre, it's like getting a lottery ticket where you have a very low probability of getting less than 2x on your investment. 500 million, right? But the sky's the limit on the other side, 60, and that's our first product. Then we do hepatitis B, HTLV, human papillomavirus, herpes, all off that same platform. Maybe the monogenic part of our platform works out. We're doing phenylketonuria. That looks really good. We've got results in mice that show that we can cure the human condition with the human gene. And so that has high probability of moving forward and getting into the clinic. And then immuno-oncology makes all that stuff look like a drop in the bucket. But don't ignore that other stuff, by the way. Monogenic diseases are selling at high prices, too high, okay? The SMA cure for $9 billion, I think even Novartis would probably admit internally, they probably paid a little bit too much. All right, for that, recouping their money, unless there's a platform behind it, is maybe tough, especially given that, like I said, there's a lot of ways to do the same thing in software. So another solution may come along that's better, faster, cheaper, right? So they may not get 19 years just because they have 19 years left on the patent. So this is an important new, sort of factor in pharmaceuticals. But let's go back to monogenic diseases for a moment. There's 7,000 of them. 1 in 10 people has a monogenic disease. The treatment of monogenic diseases is responsible for $2 to $4 trillion per year of palliative treatment to keep people comfortable until they die. That is a juicy market for pharma companies. Goldman Sachs, anybody here heard of them? They predicted this industry as $5 trillion. I think they're low. There's way more than $5 trillion worth of stuff to unlock, value to unlock for people in the gene and cell therapy revolution. Way more than $5 trillion. Pharmaceuticals today total out at just over $1 trillion per year. It's a whole new world of opportunities.
Can you speak briefly on a little more on the trial design and timeline?
Sure. The question is on trial design and timeline. David?
So as you saw, it's a 2-part phase 1. Right now, our manufacturing capacity is 2 products per month. And so the total phase 1 is 18 patients. And so we have some staggers in there about the dosing. So we've proposed to the FDA, and they've accepted this, a kind of unique way to handle the schedule. So we can just continuously enroll, make products, and freeze them, and then dose according to the schedule that the FDA has requested. So we can maximize our manufacturing capacity while obeying all the safety features that are required, right? Because till it's in the person, it's not a risk. So we think the trial, from trial initiation to end, is about 9 months, then about another 3 months to finish the last phase 1 follow-up on those individuals, and then that wraps the phase 1 study. Then, as you know, in gene therapy, people automatically re-enroll in a long-term follow-up study, 15 years, and those follow-up conditions are prescribed in advance by the FDA. We We don't generate those conditions. They're just what we do. So they'll be in the 15-year follow-up after that. But that's infrequent, and there are really no study endpoints in the 15 years other than safety.
Let me just add one word to that. It's an open label study. So soon as we get patients that have been treated, we can start to report adverse events or lack thereof. So we may get safety data pretty quickly on this. My hope is that March, April timeframe, we may get our first patient being infused. And so, think about June, some safety data. Now, what happens after that? Well, we don't prescribe a treatment withdrawal, but if there aren't adverse events from the treatment, and if people feel better, the doctor and the patient can agree without, any violating any of the FDA rules or our trial to go ahead and stop their antiretroviral therapy. So it would be informal and it wouldn't be official in terms of a phase 2A. But you may start getting some information sort of late summer next year about some informal efficacy readouts. So that's my prediction. I think it would be reasonable to think that by the end of the year, there may be some excitement in the industry if these patients do get a durable response, and that we may see some of that even though we haven't started the phase 2A. Now, soon as we start treating some patients, we will go back to the FDA and discuss, can we re-enroll with a treatment withdrawal? And we'll see what they say. The FDA is somewhat mysterious in some cases, but we have also found that they are advocates for new solutions for patients, so they do have an incentive to try to do this stuff with the limit of safety. So they're on the team in terms of keeping people safe, which we are very concerned about as well.
Just to follow up on the trial design, what are the primary and secondary endpoints that you're going to look for in safety and then space to be?
So repeat the question.
Yeah. So the question was asked to describe the primary and secondary endpoints in the phase 1 study. So the primary endpoints are all safety related, of course, and the secondary endpoints include some defined immunological assays and then a grab bag of other virological and immunological studies. So the defined assays, really we think that we want to show that the patients retain this elevated CD4 T cell response to HIV gag protein and that those cells are not eliminated. Just as an aside, we know in HIV that there is an ongoing process of birthing these cells from the bone marrow and destroying them. So even in the presence of what we call suppressive therapy, there's a pathology that continues. So we want to make sure we don't fall into that trap, that those cells survive. We expect there to be a ripple effect of reinitiating those cells, so we should see a broadening of the CD8 T cell response, right? So to explain that for a second, right, if you look in people with HIV that have finished the acute infection, the viral sequences are all over the place because the CD8 T cells are attacking infected cells and trying to kill them. Once the CD4 goes below a certain level, you can't make new CD8s. The virus escapes the last one you made, and then it just sits there as an infection, and the sequence doesn't change anymore. So the sequence is stable, and that's the absolute telltale that you've lost that CD8 response. We'd like to see that CD8 response broadened as evidence that there's new clones being activated and the capacity to keep up with virus variation is restored. So those are two very important ones. We will look in the other things for a whole variety of events that we're interested in. We'd like to be able to measure this so-called virus reservoir. We don't put it in a defined endpoint because we don't think any of the current assays really stand up. They're not going to give you significant data in a trial of this size or practically in a trial of any size. But we think we need to stay on that. We need to keep working with academic groups who are worried about this, keep refining that capacity to look at that part of the problem. We'll look at antibody levels, and we may ask some other reservoir distribution type issues. Other people have used sampling of the bowel tissue, sampling of lymph nodes in vivo. I would not think we need to go that far. I think the markers that we can get from blood sampling probably will sustain us.
Is that the way going forward also for claiming the cure or claiming the
No, I think that everybody in the field agrees that cures occur when you remove standard antiretroviral medication. And then nobody agrees on how long you need to be able to suppress virus off medication in order to make the statement, right? So that number is very much in flux. It's completely undecided in the field. And as you can imagine, we don't have something like in HIV, we don't have something like the 5-year cure, like you have in cancer, right? 5-year remission. You really can't build criteria for defining these things until you actually have events to study and to use in the definition. So that will evolve, but we would really like to see months to potentially year of remission, of stable virus suppression off of therapy. The person, Matt Chappelle, that you saw in the video, Matt received the Sangamo product. He is stably off meds with a viral load of about 50 to 100, persistent. So if you understand HIV business, that's dust of HIV down there. It's just barely detectable. And you have quite a number of people, let's say a few percent, 5% maybe of total HIV infected people who fall into the category of long-term non-progressors who have stable viral loads like that naturally. They never take antiretroviral medication. I have personal friends that are 25, 30 years in this condition. And so that looks like a fairly stable situation. And to tell you a dirty secret, they have tons of GAG-specific CD4 T cells. So,
if I could add one thing to that, the key in my mind is that the scientific community look at some of these measurements and get excited about the result. I think that's what will buoy my confidence one more level. Now, the other thing is if somebody does decide to drop off of their antiretroviral therapy, normal rebound to full viremia is 21 days. So if they at least share that information with us, we can measure their viremia, and the curve of that would be very revealing. If they are able to stably suppress it after 21 days, that's a big deal. And if we can look at that curve, we can understand how the CD4 cells and the resulting CD8 cells took over for the antiretroviral therapy. I'm not somebody that can evaluate these things, but I can tell you that these two guys can, and all of the people on our scientific advisory board and folks that we're collaborating with at NIAID now would be able to say, yeah, compared to everything else we've seen, this is on the right trajectory. I think that will be a very motivating moment for the industry.
Will you follow this up, the prophylactic version for the label purposes, because you are effectively treating HIV, so in a prophylactic version, preventing HIV from infecting.
I'm going to try to answer that, and then I'll let David give you more detail. So I'm just going to answer it from sort of the CEO market perspective. At $500,000 a cure, it is not worth it to start going out there and treating people prophylactically, and you might as well wait until they get it, right? Since you can go ahead and once you detect it, you can get them virally suppressed. They have all the incentives to do it because nobody wants to die of AIDS. And so when you tell them, hey, you're going to feel like crap, but it's only 1 to 3 years and then it's out of your life forever, they're all going to get on board. So we'll stop the epidemic growth of this by early detection, early treatment. It's something that will be sensible out there in the environment. Now, prophylactically, what price does it have to be at? That's the question. When you tell me that price, I might be able to predict how long it'll take us to get there, all right? Because this is a very sophisticated modification of the immune system that renders you immune. Now, if you had $500,000 to spend, could you take this prophylactically and then never worry about HIV again? Probably, right? If it works for the patients, it'll work for healthy people. You have great immune systems. If they've never been challenged by HIV, you're all set for this treatment. Day one, you don't need to go on ART. you can just go ahead and come in, get the leukapheresis, and a month or two later, you're immune for life. So it can be used prophylactically, but it's all going to be about economics, about when it gets there, and then, maybe it'll make sense. Now, there's other ways we can go with this as well. There's nothing that says that there's not a gene and cell therapy approach to better enable vaccine strategies, right? If that happens, I think it may happen at our company as well. So, otherwise Gilead's going to ask for that project too. But the reality is that, there's a lot of things that you can do to not just make your body operate better, but you can go ahead and you can impact other drug therapies through gene and cell therapy. And that may be the way to do this prophylactically. It may be to take a failed vaccine, right, and then to add something to it that would bring it over the line where you could expect that you'd get enough people that were made immune by that strategy that you would end the sort of herd infection, or sort of the normal spread of the virus within certain communities. And that would be incredibly valuable, and it's certainly something that's on our radar screen, but we don't have a specific solution to it that we can talk about right now. David, did I say anything incorrect, or that you want to add something to?
No, I would make a really tiny comment, which I filed on that 2.5 years ago. And so it's a really good idea. But cure and prophylaxis, has been a very strange issue in HIV. So David Hardy mentioned it, taking these antiretroviral drugs. Bill talked about it too, for preventing infection. So in organized clinical studies, this is about 90% effective. In community use, it drops below 20%. And it is a very large healthcare money ante to get into a PrEP type prophylaxis. There's a lot of screening that has to be done. There's a lot of medical maintenance to follow up. That's because these are lightweight chemotherapies, and these are not simple drugs. You heard a lot about that. But there's a core of people who are seeking prophylaxis no matter what these hurdles are. And whether those people would want to engage in something like this, I think it's a realistic possibility.
Actually, a question for David. I think Dr. Hardy compared the T cell versus the CD34 positive cells, and
Right.
by targeting the GAG-specific T cell, does that do anything to the durability of the survival T cell and also the durability of the treatment effect?
So we've been looking at that in just the normal product processing, and also we had to do, as safety studies, we had to do a lot of humanized mouse experiments where we were injecting genetically modified cells into these immune deficient mouse models. We find no aberrancy of the genetically modified CD4 cells at all. They do everything you expect them to do. They don't do anything you don't expect them to do. We've even done this analysis called repertoire analysis, where you sequence all the T cell receptors in a population and you ask how many of those respond to GAG, and the numbers all fit. They look like a normal immune response. Once you allow them to sit around for a while, they go back into memory and they just stay. So we don't see any real issue with that. Really, the number of times when these CD34 positive cell modifications and bone marrow transplantations are going to be done is very small, and a lot of these studies have failed to complete enrollment because there's simply no imaginable way to do this in people unless they have a cancer diagnosis.
And for the study, are those patients going to receive conditioning prior to infusion?
Yes, and this conditioning is extremely low dose of cyclophosphamide, and that's what David has experience with this. And we talked to him before to get insight into this and to understand this. Many people have a fear of cyclophosphamide conditioning, and we give one dose at a 0.5 gram per meter squared, just to give you the number. In clinical cyclophosphamide use, when you're trying to suppress cancer or trying to eliminate the bone marrow, you're talking in 1, 2, or 4 grams per meter squared given repeatedly over maybe 5 to 7 weeks. It's absolutely no comparison between our conditioning regimen, which is just that little tincture almost, to drop the cells. And it's an interesting mechanism of that. You know, you reduce the cell number a little bit, but those low doses of cyclophosphamide give an unexpected boost to immunity. And the reason is because the most labile cell that's eliminated first is the T regulatory cell. And so then you get this burst in growth. And that's probably how it works. You get rid of the T regs, you put these cells in, there's nothing to prevent them from growing, so they just proliferate normally and engraft at a high frequency.
Do we know any impact on patients' fertility from those molecules?
Absolutely none. And in fact, there are published examples where this low-dose conditioning has been given to women who were unknown to be pregnant at the time, with no effect on the fetus, no effect on the delivery. Of course, the high-dose cytotoxicity for high-dose Cytoxan you cannot give to pregnant women.
So the transduction efficiency, how does your ratio compare to Sangamo, the range of modification that they were able to achieve in the study?
So the numbers, and remember the lesson in David's presentation, that you had to get a so-called biallelic deletion. If you knock out only one gene of CCR5, the other gene makes enough CCR5, the cell's still susceptible to infection. So Sangamo got a modification in about, let's be generous, about 25% of total CD4s in their pool got a modification. Less than 10% of the cells in the pool got both alleles knocked out. So it's the bulk. So out of the bulk T cell population, you really only had 10% with CCR5 knocked out. In a bulk population of CD4 cells, 10%, never you see 10% making CCR5. So the fraction of potentially CCR5 positive cells is only a small fraction of that. So, the modification, which is quite difficult, has a lot of cell loss due to the use of things like adenovectors, which are kind of brutal on T cells, and then there's these difficult systems for editing. The T cell loss is substantial, and they had to grow the cells a long time after the editing event to get back to a normal dose. So the fact that they saw something with something that's really kind of a residual product compared to ours is what gives me encouragement that we're on the right path and that we're doing something that's substantially improved over that, learning from them. And I don't criticize them at all. They were state of the art at the time.
You had a question?
Yeah. Can patients develop an inhibitor to the vector that you use? I know in hemophilia gene therapy, some of the adenovirus vectors, some people have resistance to.
Yes. So the question is, if you're going to use vectors like adenovirus vectors or adeno-associated virus vectors more correctly, can people can be resistant to them by prior exposure to that virus? Can they be resistant to this virus? And if you've had a gene therapy before that used a lentivirus vector, you could potentially be resistant. These lentivirus vectors are what we call pseudotype. They don't use the HIV envelope attachment protein for cells. They use one from a different virus called vesicular stomatitis virus. And that's a good immunogenic protein, but very few people have natural immunity to VSVG. It's the VSV protein. It's extremely rare. And so the resistance to it is also extremely rare.
Would you test the patients first to see if they had resistance?
Probably not. We've just never seen it as an issue. If the issue arose, there are many, for the technical term, serotypes, right? So you have many different serotypes of VSV envelope protein that can easily be substituted with no problem.
Can I add one thing? And again, David, I'm going to rely on you to correct me if this is wrong. One of the aspects of lentiviral vectors and the types of treatments that we're doing is that it's at very, very low doses relative to what you typically see in AAV. One of the reasons that that's a bigger problem in AAV, is that the doses can be 10 or 100 times greater for one patient. Well, now you're talking about a fairly massive immune response that might give them a latent effect that would go ahead and attack the next treatment. You also have AAV, is not an enveloped virus, or a membrane virus rather. So as a result, the natural forms of AAV that have been discovered and utilized as vectors exist out in the population. So you do need to look and see, maybe somebody's caught the wild type version of AAV9 before you try to treat them with AAV9, right? But as David was saying, very few people, maybe none, have already seen VSV. And so you wouldn't expect it in the first treatment. The question would be, okay, can you retreat them with the same vector? Probably. It's just going to depend on, how much of it their body saw and how much it reacted and how much antibody production, against that particular virus is there.
Let me just say something. Having 30 seconds to reflect, I think I thought past your question a little bit, right? Remember that the vector itself encodes nothing, encodes no proteins. The only thing it encodes is those microRNAs. So once you make a transduced cell, there's no source of any protein. The only place the VSVG protein came in was in the vector, and that all occurred outside the body. So, if you had, we can spin castles in the air, but there's really no realistic way that immunity can block the ex vivo process. It's a different story if you start to build an in vivo process, but then that's the in vivo process is more appropriate to the question you asked. And all the AAV treatments that you're worried about are all occurring in vivo with basically ocean liners full of AAV, right? Just gallons of AAV. So the challenge to the immune system is extraordinary in that regard.
Any other questions?
This is just for clarification. If a patient's on ART and then they go through your treatment, do they still remain on ART?
Yeah, the goal. So the question is if somebody is on ART and they go through our treatment, would they still remain on ART? So for the first human trial, we keep them on ART for a safety reason. So we'll study their blood and we'll try to determine, is it working, without requiring them to go off of their ART. But the goal of the project is to reduce or really to eliminate ART.
Okay. If they remain on ART, what's the incentive to go for your treatment versus just any other drug?
Well, even reducing it would be a big deal. But you saw that one of the HIV advocates, Matt C., I guess he's well known, so he's been off antiretroviral therapy for a while. The same thing's true about the patients that received bone marrow transplants, Berlin, Dusseldorf, and London. So yeah, that's the goal. We think it's achievable. We may not demonstrate it in the phase 1 or adequately, to our taste, but I think we should get some sense of whether that's going to be possible, and it's absolutely the intent, and I think it's a high probability of this project.
And then if you get good phase 1 results, would the phase 2 just be like a registrational trial?
Well, I think we'd probably enroll more patients, so we wouldn't just re-register to the existing ones if I'm,
No, I think it's realistic in this kind of situation to register out of phase 2 if the phase 2 is appropriately powered. That would mean that somewhere along that line, either in a 1B or in a non-IND controlled study, like an IRB study in the university, we could then do a treatment withdrawal, a structured treatment withdrawal. And we would like to see the data to ask ourselves if we can make some predictions from the data if there's a stratified group. Then maybe there's a group that we would say, these people have the highest possibility of being antiretroviral free, or if we can't make a stratification, then maybe we just make the argument that everybody can go into a treatment withdrawal study according to their desire. The recent research says there's no long-term health consequence of going through a treatment interruption under a supervised situation, right? Non-supervised is different. If you do it in a supervised study where you're being monitored intensively and you go right back on your regimen as soon as you have virus rebound, then that's no risk to your long-term health, no risk of developing a resistant virus.
The purpose of all HIV cure studies is to ultimately control HIV without antiretrovirals. And so a new term has been coined called an ATI, an analytical treatment interruption, which is what is being built into most, if not all, cure studies. One of the things that is being encouraged, though, in these is to have some sort of laboratory marker that you can quantitate to say, since this intervention that we've done, is this marker demonstrating something new that wasn't there before the person got the intervention, that gives us assurance that we have in fact impacted this person's immune system in some way, that an ATI would make sense. Historically, ATIs were done just based upon the idea that they were a good idea. Let's see what happens. You know, we cooked it up, let's see what happens. But so many ATIs have been unsuccessful. So the responsibility of the researcher, of the company, is to prove that something has changed, that there are more, for example, GAG-specific CD4 positive T cells that are expressing the new gene put in as a marker to say, okay, we did the first step, now let's try the next step. But ATIs are going to become something that is really the proof in the pudding, because unless you stop the medication, you really don't know. And the other thing is that oftentimes having the viremia come back, at least temporarily, may in fact trigger the activity of whatever HIV mechanism for protection you put in. Because what may happen is the cells that are protected will continue to live, the cells that are not protected will die. And it'll be a process over time where those that are living will expand in larger and larger numbers, and those that were not protected will go away. So that eventually the entire immune system will be full of protected cells. So it actually may even have, the ATI may even have a, not only a proof, but also a role in triggering what may need to happen.
So, why don't we take one more question, and then I think we probably should wrap, because I think we're going past our original time limit. Did you have one more? Sure.
Just a follow-up question on the discussion that we just had now. So why ATI was not incorporated into the study design? Because one, it gives you more evidence that your therapy is working, and secondly, it actually triggers you stimulate the immune system to help fight the HIV better. Why you didn't incorporate that into the study design?
So the question is, why not incorporate an ATI into this initial study? David?
Yun, the winds blow differently at the FDA on different days, and so the answer is two-part. One, me personally, I think that we need to be cautious about this. ATI is not something I take lightly. And second issue is, when we were developing the protocol and doing the original interactions with the FDA, that was a time when the wind was blowing against mandatory ATI. Now the wind is changing a little bit, but you can't keep rewriting this program every day. So we think we're in a good spot. We took the more conservative view, but we also know that, like I said, you can come off of this study, go into an IRB-controlled study at the university, which is not under the mandate of the FDA, because you have no intervention. You're just removing something. And so that's quite possible to do. And we're talking to NIAID about them taking over the intervention study, because they have the ATI part, because they have a lot of experience about intense monitoring during those treatment interruptions. And I think that's going well. We're working very closely with them now on product characterization, on a number of other issues, because this product's not only looks like great for clinical, it's a load of gold for researchers in this area.
Yeah, that's why as often as you can't make it mandatory. You can't go into the study and have people off medications a mandatory part of the study. So usually what happens is they may modify the study to make it allowable. it's still a decision between the person who got the cells and the investigator. You can't make someone stop their medications. That's the hard thing.
Okay, so we'll hang out if you want to talk to us informally, but that'll be the end of our formal program, and I want to thank our scientists for great presentations today, and thank you for coming.